millions of compounds are synthesized, that biological activities have been determined. Right, all these informations are stored. Right, similarly, you have a biology side. The proteins are evolving. You have so many structures of protein. Right, and then how these structures, how these molecules are interacting with the ligands and so on. There are huge quantity of volume of information is generated. So using this, the molecular modeling is computation, visualization, statistical understanding are the input, and then molecular data management. Everything put together, you call it as the molecular modeling. Now, what are the inputs? Where we get the experimental details, and see, basically, you get the structural bioinformatics. We are all familiar, or extra structure determination, NMR. Right? This gives you some sort of a structural information about the protein or DNA or ligand, small molecule that you handle. Then QSAR, quantitative structure activity, or quantitative structure property, or quantitative structure toxicity, right? All these are really useful in the rational drug design. Now, I said in one hand that we need to have the three-dimensional structure of the proteins or the biological macromolecules to understand the interaction process. If those structures are not available, only the sequence are available, then we can use the input from the bioinformatics, basically the homology modeling, right? So use the homology modeling and try to get the model structure. So you apply the principles of bioinformatics, also chemoinformatics, because you know that the chemical space is very vast, huge volume of data. Chemical space is really large, therefore we need to exploit the chemical space. And then in the exploitation of the chemical space, you need the combinatorial chemistry, wherein you will have the chemical similarity and chemical diversity. Then what are the mathematical techniques? I already mentioned to you that we have the quantum mechanics and molecular mechanics, which is based on the Newton's law, or any one of the classical physics-based variations that can be applied here to understand the interaction process. What are the basic modeling strategies that we apply in drug discovery? Right, this can be a ligand structure. The ligand structure sometimes known and unknown. If you don't have the structure, then we need to generate the 3D structure, similarity and dissimilarity, homology modeling, and then you use the high throughput screening, combinatorial chemistry. And then known as, well, if you know what are the information that you need to know, its active site at which you are trying to interact the protein with the ligand, active sites in the protein wherein you want to interact the ligand, or receptor based drug discovery, de novo design, 3D searching, and so on. In the case of a known category, it is really easy. You can understand this is the indirect drug discovery or ligand based DD. Analog designs, 3D QSAR can be used here, 3D or 2D, and pharmacophore mapping. Pharmacophores, I will come back to what are pharmacophores. And if you don't know the receptor structure, ligand structure, and if you don't know the receptor structure, you can use the 3D. If you know the receptor structure, then you can use the principles of rational drug design, and you can understand this quadrant very easily. Uh, in the first quadrant, we talked about the unknown of the ligand structure and known of this receptor, and rational drug design essentially based on the structure based drug discovery, wherein you can apply the molecular docking because you know the structure of the protein, you know the structure of the ligand, you can put them together, look at all the interaction feasibilities. So this slide basically gives you the information about the protein and the receptor, how these two are interacting, what are the techniques that one can use to understand this. From computational point of view, I describe here, I already told you molecular mechanics, which is essentially based on the classical physics, and quantum mechanics, you know about the Schrodinger equation. And in quantum mechanics, I may not go into the very details of the quantum mechanics aspect, but I'll just give you a flavor of what kind of electronic structure theories are used today. When we started it, we used to talk about the semi-empirical and then ab initio. And today people are talking about the density functional theory, wherein you can handle more number of atoms. And in the case of a molecular mechanics, it is totally a empirical functions, right? And these empirical functions are capable of looking at the molecules or atoms in the molecules. As if you say a molecule, then it is the molecules are connected by the atoms with a bond, and these bonds can be considered the elastic bond. This will have a elastic spring kind of a interaction. Within the limits of Hooke's law, you can understand the interaction between these two. The potential energy of a molecule is described by a mathematical function, what we call it as a force field parameters. Force field parameters are used to describe the interaction potential energy of the molecule in the three-dimensional space. So this is what happens here generally. In fact, this received a Nobel Prize about 3 4 years back. All right, wherein the merging or marrying of marriage of classical mechanics and quantum mechanics took place with the help of the famous scientists. So they used both the principles of classical mechanics and quantum mechanics to solve the problem. Right. So this is called the mixed quantum classical mechanics methods, and then there are a lot of techniques are there. I don't want to go into the details. Traditionally, we use quantum mechanics to understand these small molecules and use the classical simulations for the large proteins and protein ligand, DNA protein, DNA ligand kind of interactions. But if you know that there are a lot of limitations of classical mechanics, when you take the protein ligand system, the interaction process right requires much more rigorous fundamental treatment. Then if that is the case, then only those regions which are interacting with the protein and ligand can be treated very high quality level of theory, maybe quantum chemistry based electronic structure theory or density functional theory kind of a representation can be given to the ligands and the active site of the protein. So this is called, you can use a mixed quantum and classical mechanics to understand this. Here is the idea that you can very clearly see, that you can see that a large protein is given, and then one another level. I think I put two circles. The interior one will, the interactions will be very very important, and the outside one is the interactions are not important, and then it's not that important like the inner core, the second level of treatment, and you have the third level of, so three layers I can split. Right, the internal one I require more rigorous treatment, that's a chemical active part that will be treated by the quantum mechanics and the interface region, and then the large environment, and then you have a solvation also. All right, all, suppose you consider this protein and it is solvated, so you have a solvation also. So all these things takes care of the interaction. Therefore we need to when you do the modeling, at what level you want to do the modeling, right, that you should decide, right? It comes by experience. You should decide whether you have to apply the rigorous quantum mechanics, the next level of molecular mechanics, or third level of coarse grained kind of approach, wherein you certain groups are modeled as a beads and things like that. Therefore you have levels of treatment, levels of modeling. All right, so you have a very rigorous levels to the classical modeling is possible. So here is an example. I hope you could see the video. It's coming up, right?
Yes.
Hello.
Yes,
Yeah.
sir.
All right. So here is a realistic simulation of a molecular dynamics. All right. The generally when you handle the protein ligand or DNA ligand or protein DNA, these kinds of biological system or any other chemical system wherein the dynamical information is fundamentally very important. All right. How a particular object or particular molecule interact with the protein or interact with DNA, how with time evolves, right? So that's the one. You can see that a particular, I have a some protein, all right, in which some ligand molecule is trying to interact or diffuse through that particular medium, all right, by creating a way for it, all right. So this is a dynamical picture that is shown here. All right, large number of atoms or molecules are allowed to interact in this particular time, period that you are looking at it. Therefore, all the interactions are important. All the movements of the atoms are important. All the movements of the group of atoms are important, right? Therefore, at in all the instant of time, you save all the coordinates, all the displacement, the velocity, right? Everything will be saved in the molecular dynamics simulation. Therefore, you require huge computational power. And more than that, you can see that the certain vibrational motions of these molecules occur at the time period occurs at 10 power minus 14 second. Therefore, if you need to solve the differential equation for looking at the interaction picture, right, the time that you use to solve the interaction equation is 10 power minus 15 or 1 femtosecond. Right. Therefore, if you want to look at any process related to the protein, which is taking place, let's say millisecond 10 power minus 3, my starting point will be at 10 power minus 15. Therefore, 10 power minus 3 divided by 10 power minus 15. Therefore, it is 10 to the power 12 times I need to solve the equation, right? Or that much of information I need to store in terms of the XYZ Cartesian coordinates of each positions of the atom, of the protein, of the ligand, and their velocities, and so on. Therefore, it is a huge data will be stored in the computers, right? So that's very, very important to understand this. Therefore, the from 1970 onwards, this molecular dynamic simulations have been popularly used in biology and chemistry community, right? And as I said in the beginning, that we have two kinds of approaches. One is based on the quantum mechanics, another based on the classical mechanics, and the marriage of both quantum and classical mechanics, right? Today, we all handle everything with the help of large systems are handled with the help of the classical mechanics-based approach. What are the characteristics or what are the nature when you do the classical MG? The degrees of freedom or number of molecules. The idea here is you have the atom, the atomic. It is everything is dealt in atomic. It's atomic level interactions. All right. So you look at in terms of the solute and solvent. Equations of motion is classical dynamics motion. It is Newton's equations of motion. And what is the governing theory? Because you use a statistical mechanics, which is a classical statistical mechanics. You apply the principles of classical statistical mechanics to derive certain aspects of properties. And in the case of a quantum, you use a nuclear, you use the electronic and nuclear concepts. Right, electrons are handled very nicely. In the case of the classical, there is no bond making and bond breaking, but in quantum, you have the ideas of bond making and bond breaking, and you use the quantum dynamics and quantum statistical mechanics is used. Here, this slide very clearly describes you. All right, I'm sorry for the clumsy slides, but the classical approach, you will use the F is equal to ma equation. All right, in the case of a quantum time dependent HI equal to ESI equation that you will be using. But you look at the F is equal to ma equation that this acceleration, in the case of a force, ma gives you the force. But what is acceleration? Acceleration is rate of change of velocity, and velocity is rate of change of displacement. Therefore, force is equal to mass into m d square r by dt square, where r is the displacement. So this can be written in the form of differential equation. What about the force? The force is nothing but in a conservative system, force can be obtained from the derivative of the potential energy. Therefore, dE by dr will give you the force on the system. Therefore, this combining these two, you get the differential equation. This differential equation can be solved. Starting the integration step is numerically solved. The integration step is 10 power minus 15 second. All right. So these are the fundamental ideas here. Let me not spend time. The quantum one are more advantageous in the sense that it is more accurate, right, from theory point of view, but it is really time consuming. Instead of that, we can use the classical approach to get to the large scale simulation. Basically, for biomolecular system, classical simulations are widely used. And where we apply the classical simulations, you have the, you can look at the RNA, DNA protein interaction, protein ligand interaction from the point of view of drug discovery. What are the input that you need to give? You need to give the position, that is the coordinates. Where do we get the coordinates? You get the coordinates from the crystal structure details, and then you can apply the statistical principle, and you can get the charges. Generally, charges and force field parameters are basically derived from quantum mechanics, and you use the idea of similarity and port those details to the large scale systems. Basically, bond information, that is important, how these two atoms are arranged in the three-dimensional space, or in the case of a protein ligand complexes. I'll explain to you. This is what we generally look at it when we move from the potential to dynamics. We have the potential. Potential is a mathematical expression which describes all the interaction process involved in a classical molecular dynamics. To run the simulation, we need to get the force, and I have already explained to you this equation. F is equal to ma, and m is the mass, and a is the acceleration. Force is equal to minus del v. Del v here represents the potential energy of the system. In a one dimension, this can be written as minus dv by dr, and mass into d square r by dt square. This is a second order derivative here. This is the first order derivative of the potential. Right, these two are connecting here. Right, this equation is numerically solved to get all the interaction details. But the interaction details are given in the term potential. Right, the potential is explained here, V of r. It is a function of the coordinates. This can be split into a bonded and non-bonded terms. These are combined together. So the non-bonded terms are the van der Waals interaction and the electrostatic interaction. Van der Waals forces are, you know that they are described as the by Lennard-Jones potential, or any, there are several van der Waals potentials are there. Generally, in molecular modeling and simulation, or in any dynamical simulation, we use the van der Waals one, and the van der Waals one is the Lennard-Jones one as a van der Waals one, wherein you use a 6, 12 potential, A divided by r to the power 12 minus B r to the power 6. That is a potential one uses for the Lennard-Jones. And electrostatic. Electrostatic can be treated as a charge-charge interaction, or charge dipole-dipole, or charge dipole, and so on. The computation, right, in the mathematical expression, the computational time required to do the calculations increases. Right, the van der Waals interaction generally arises mainly because of the induced dipole-dipole interaction. If you have the permanent dipole-dipole between any two systems, the permanent dipole-dipole interaction varies as r to the power 3. But when you have the induced dipole interaction, then it varies as r to the power 6. It is an attractive interaction, right? And you have a repulsive, A r to the power 12 is the repulsive. That is, you have two atom A and B, and you are trying to bring them together. You can't push them beyond a particular point. Okay, the optimal is the bond distance. Beyond that, you push them together, then it repulsive. The charge cloud will repel, therefore the system will break. Therefore, there is optimal distance, right? That is called that distance is determined by what is called the van der Waals radii for a particular atom. Electrostatics, in a simple QA, QJ, RIJ, that is a Coulomb's law. We are familiar with the Coulomb's law. You have two charges, the product of two charges. The force or potential between the two charges is directly proportional to the product of the charge, and inversely proportional to the distance. In the case of a potential, if you have the force, it is directly proportional to the product of the charges, and inversely proportional to the square of the distance between them. I'm sure it is very clear. Dear participants, things are clear?
Yes, sir. Yes, sir.
Yeah.
Sir, I think you are mute. Please unmute yourself. We can't hear you now. Subramaniam, sir. Sir, you are on mute. Please unmute yourself.
Yeah, I'm sorry, something happened. You could hear me?
Yes, yes, sir.
So the non-bonded is van der Waal interaction plus electrostatic interaction. Van der Waal interaction is basically modeled with the help of the 612 potential. Electrostatic interaction is modeled with the help of the QA QJ divided by RJ, that is essentially Coulomb's law. In the case of a bonded terms, you have a bonded is, you can look at, you can see the animation here. The a particular group is stretching, it is compressing and elongating, compressing and elongating, and you can see that a group is bending and unbending, and your group is rotating, that represents. So the stretching is one energy term, it is bond stretch. The angle bending is another one that gives energy to the process, and you have a rotation, that is a torsional or dihedral contribution. Therefore, the any molecule in a three-dimensional space can be represented in terms of the bond length, bond angle, and torsional angle. These are r, theta, phi, or the angle. With this, all these involves energy. Therefore, this can be this can be represented. All these can be represented in terms of a mathematical form. Alright, these are the mathematical formulas for that bond stretching. You can see that a stretching is happening. The bond stretches KB into B minus B0 square. The summation over all the bond pairs. And you can see that the angle is bending. Therefore, when have a three angles, three atoms are connected, 1, 2, 3, it involves that angle, that theta 0 is the equilibrium 1, and at any instant of time it can take any value theta, therefore theta minus theta 0 square. Therefore, this involves energy, that is k theta into theta minus theta 0 square. This kb is called the force constant. K theta is also force constant. And k phi, it is also energy, it is a rotational energy required to rotate the particular bond. Therefore, you have several variations of these expression. Essentially, the stretching won't change, bending won't change, but the torsion, there are different kinds of torsion potential. But essentially, there are sines and cos of the rotation angles. All right. Therefore, these are the mathematical expressions used to compute the energy of the systems. Then how do you integrate? You can use various algorithms. I'm not going to the details. You can integrate the equations. That is the equation which I told you, dv by dr equal to mass into d square r by dt square. That equation can be solved numerically, and you can understand most of the phenomena. And one important concept is two concepts. One is the periodic boundary conditions, that is, see, you do the experiment in a certain conditions, experimental conditions, where at any instant of a time the given molecule will look at similar kind of a molecule in the three-dimensional space. Therefore, you need to have the periodic imaging. You can see that a particular group is coming in and particular group is going out, but the number of a particle in a particular cell is always constant, right? It depends on what kind of ensemble. Ensemble means collection of a particle. So what kind of ensemble that you are trying to use and then model the system, right? It is defined by the periodic boundary condition, right? And you can see that in a particular group, how the various groups are in a realistic conditions, how the various, you can see that the loop, and you have a a core structure. The core structure is not doing much change, but the loops are making quite a lot of change. All right, these information one can understand from the visual understanding. So these are the basically techniques which are very well practiced in the drug discovery to understand the realistic information about the process. Another technique which I thought that I will share it with you here is the physicochemical properties. How do you model the physicochemical properties, or QSAR? How do you develop a property based on the structure? So you have a series of ligands, and then each ligand will have its own properties. They will have the octanol water partition coefficient log p, or how do they dissociate, or how do they binding constant or dissociation constant. Right, there are variety of properties, variety of attributes can be given to the ligands that you are looking at. So this is ligand based drug design. You need to know about the properties of the ligands. Those properties of the ligands are called the descriptor of the ligands. Pharmacophore perception, that is, I will come back to what is a pharmacophore perception. All right, and in the structure based category, we know that a docking and scoring. And what is a docking? That is, I have mentioned to you, you have a protein, you have a ligand. Protein will have the binding sites or active sites. There are several servers available in bioinformatics tools, right? So you can determine the active sites or cavities in the protein, and you can take the ligands and bind with the various cavities and you can model. So this is called the docking. And you have a pharmacokinetic modeling, QSPR, absorption, metabolism, distribution, ADME properties, you can analyze it, and database management and similarity and diversity in the system. So all these techniques, no one can integrate, apply this for the realistic modeling. Laser pointer, that's okay. That's fine. Yeah. Let me move. Right. Thank you. So quantitative structure activity. Let me briefly give you what is QSAR. You can see that biological activity can be represented as a mathematical form as a relation. F is equal to say biological activity equal to physicochemical properties. This is what here you represent physicochemical properties. That's what you put here. As a function of this, biological activity is determined the physicochemical properties. Suppose a ligand, a certain dipole moment or charge or number of rings, number of aromatic units, all right, or octanol partition, so coefficient, all these are physicochemical properties. They describe the biological activity. So in the description of biological activity, in the QSAR development, we use the physicochemical properties of the set of ligands, right, for the biological activity development. Right, this is called the QSAR. We have a two-dimensional QSAR or three-dimensional QSAR. In three-dimensional QSAR, in two-dimensional QSAR, there are so many descriptors. You have a topological descriptor, you have atomic properties can be used, physicochemical properties can be used in the two-dimensional QSAR. In the three-dimensional QSAR, you have the protein, you have the ligand, and what they do is they try to look at the molecular interaction field around the ligand, and that can be used as a idea here, that can be used to develop the three dimension, the what they call it as the electrostatic force as well as the spherical force or spherical terms and electrostatic terms, to understand how these bind with the three dimensional structure. There are a lot of work has been done in this area. right, which involves the protein structure and ligand structure, ligand field, molecular shape, distance, receptor surface, all this. So generally, when you want to develop a QSAR, what they do is you need to have the training set. For the training set, you need to know the biological data. Specifically, the biological data should be from the same group, and then you generate the biologically active conformation or any conformation which is necessary for the binding process. Calculate the descriptors. There are two ways. You can take the molecule, you can do the conformation analysis, and for each conformation, the property can be predicted, and that can be used to develop the QSAR, right? Or one can do docking and take the docked conformation, that is called the biologically active conformation, and for that you can develop the QSAR. And there are various ways of calculating descriptors. And then what is the selection of statistical method? What is the method that you want to do, whether you are doing the simple linear regression or multiple linear regression or partial least square fitting or combination of both. And then are you going to do the cross validation? How are you going to do the cross validation? All right. And these are all certain technique that one uses here, and this generate the QSAR equation, and then you have to validate this. So you have a training set as well as the validation set you need to have, and then you can predict the unknown from the development of QSAR. What is pharmacophore? The spatial orientation of various functional groups are three-dimensional features that show the biological activity. That's the idea here. That's a hypothesis. Distance, geometry based qualitative common feature, that's a one hypothesis, right? Quantitative predictive pharmacophores, from once again you have a training set with the known and unknown biological activities and use them to get the to predict the biological activities. For example, here is an examine the features of inactive small molecules and the features of small molecules, all right, which are active. Now, based on this, generate the hypothesis about what chemical groups or what functional groups are necessary for the biological function. So that you can do that. No, you can understand which functional groups are inhibiting, which functional groups are facilitating the biological function. Alright, this can be done by, for that also you need to have the biological activities, the so-called what we use IC50 values for that. So generate ligands which have the same necessary chemical groups in the 3D locations and mimic the active groups. Here there is one example, dopamine is given and it is superimposed on the other one. So based on this, you can see that certain functional groups are important, certain functional groups are not important, and all those details can be worked out here. From this, you can understand that whether electron group, electron withdrawing group, or electron donating group, or aromatic group, all right, or certain uh, structures around the particular ligand are enhancing or decreasing the biological activity. All these information can be obtained, and this also determines how many are donor, how many are acceptor, hydrogen bond donor, acceptor. All those details can be built in here. They fundamentally design what is called the pharmacophoric features. How do you discover how long this process goes? Generally, the conventional drug discovery goes up to 215 and so on. There are so many years. All right, the basic research, even today also, the chemical space is a huge space. Millions of compounds are available for a particular target. All right, but you how to reduce them? You know, you need to have a filters. All right, and then you have a clinical trials, and then um, there are various steps, and I'm sure that you are all familiar with this particular slide. I should not be repeating. And again, I may not go into the rational drug discovery, but in the rational drug discovery aspect, what we do, we have the pandemic, right? We need to identify a drug molecule, right, to solve the problem, right? So, but what are the steps here? The steps are really time consuming steps. We have seen that. Is it possible somewhere this mathematical technique can be useful to accelerate the drug discovery process? That's a question people are asking, right? Now we need not to identify. We have the pandemic now, COVID-19 pandemic we have, and we know what are all the protein targets which are important. All right, is it possible to do starting from this? No, no, it will take so many years. All right, therefore, is it what is the approach? The approach here is people use already we have a FDA approved drugs. Is it possible to make use of this FDA approved drugs by can we understand, can we have a new therapeutic approach for these handling these kind of pandemic? That's a question, right? So how do we use molecular modeling and virtual screening here to screen the FDA approved drugs? One such technique is molecular docking. People are trying to use molecular docking, all right, wherein you can quickly wanted to repurpose the purpose the drug. But in any general case, but we generally use isolate protein wherein you apply the genomics, proteomics tools to screen the millions of compounds. You use the high throughput screening. Virtual screening you do with the help of the computer to predict active, wherein the computer scientists, the computational modeling people will help them. Right? It is not necessary that always what we get from the computer is the final molecule. There are some cases where it is works out to be very positive. There are some cases it is not at all working out. All right. So this that you have to take it with that pinch of salt here, but we generally help to speed up the process. All right. And once you do the virtual screening, you select a molecule. With a molecule you can build what is called the library can be built around the particular molecule. That library can be once again used to develop new set of ligands, and this new set of ligands can be interacted with the protein and so on. So this helps to speed up the process here. Library building is really useful in the discovery process, right? So, in a essential sense, that you have one side X-ray, NMR, cryotem kind of a technique which gives you the structure, and then from if you don't have the structure, we have the homology modeling, and then you can understand how these protein and ligand are put together. Then it is called molecular docking. From molecular docking, I can go for one step beyond that. It is called molecular dynamics. From the molecular dynamics, you can screen out certain ligands which are really interacting with the protein during the time evolution. Those ligands can be taken, and taking those ligands that can be given to the instrumental group as an input, and then they can synthesize, they can look at the real assays, and then from the assays you can identify the candidate. From the candidate, what you can do is we can go for clinical trial and so on, drug to market. But most of the time it fails. If it fails, why it fails, that can be understood with the help of the molecular dynamics. If it fails, how to knock out a particular group which is inhibiting it, how to improve that, and all that can be once again, we can use the molecular modeling technique to improve that. So these are the certain aspects of molecular modeling aspects here. I'm not spending time on this. How long I have, please? How much time I have?
We are 200.
Hello?
Sir, we can go ahead for at least for 15 minutes we have, sir.
15 minutes. I think I will finish it.
Okay, okay.
Yeah, this slides I will skip. All right, I may not go into the details here. Yeah, as I told you the beginning, docking is one technique that people are today. I'm sure that all are familiar with the docking. Many have attempted. In fact, the hackathon, in the drug discovery hackathon announced by the government of India, I'm sure that many would have participated in this, right? Many have used docking tools to understand that. So it is really a very complex problem actually, but it is made very simple. Protein has 3 degrees of freedom. Similarly, ligand also 3 degrees of freedom. And when you put them together, it is a 6-dimensional problem, right? How to fit these two together is the important one. This can be achieved by variety of ways. We have what is called rigid docking, wherein both the ligand and protein are considered as rigid entities, and you have a semi-flexible docking, That is the one of the molecules, that is the ligand is flexible, other protein is rigid, and both are flexible. Actually, in a realistical sense, both the protein and ligand are flexible. But no, all these are depends on the time. uh, there are a lot of technique. The Schrodinger came they came out with one technique called the induced fit technique, which model the protein in a protein and ligand in a more flexible form. There are variety of programs are available. You have a Glide up to Autodoc and Autodoc 3 and so on. And so these are all some summary about the performance of various docking tools. Okay, it depends on the availability of a particular tool, and then you can look at it. Um, so this is the technique that we use here actually in our group. All right, I'll skip all this. I will really go to the example which I want to show you. So let's take the SARS-CoV-2, and you know that these are all very important protein in SARS-CoV-2, the membrane protein, and envelope protein E, spike protein S, and then nucleocapsid protein. Structure of the SARS-CoV-2 is like this. And you analyze more, we know that these proteins are very some of the proteins which are very important in the case of a SARS-CoV-2 is, right, I don't know. As you said, I'll try to complete. So what we have taken is, we have taken SARS-CoV-2, important protein, the main protease, RDRP, RNA-dependent RNA polymerase, spike protein, and the human, the proteins which are involved in human pathways, that is the TMPRS2. This we have taken in our group to model certain class of molecules, especially the approved drugs. Right? That result I will present. Before that, I thought that I will spend some time with various viral diseases. You can see that these are all HIV 83, HCV 89, human influenza virus, right, 1933. These are all some of the viral diseases that we have come across in the past. Now we are witnessing COVID-19. What are the classically used drugs for this? This is a 5-substituted deoxyuridine analogs have been used for the antiviral molecules. Some, and the another class is nucleoside analogs. The basically the three FDA approved drugs in this category are I have listed here. Then nucleoside analog category, we have the tenofovir, and these are all molecules which are already used by them. Another class of compounds is phosphate analog, pyrophosphate analogs, right, and then nucleoside reverse transcriptase inhibitors. This came after the HIV virus, and nucleoside reverse transcriptase. These are all some of the compounds that I have indicated here, right, for these handling these viruses. And non-nucleoside reverse transcriptase, you have this nevipiravin and delaviradine and so on. These 5 molecules are used for that. Protease inhibitors, there are several protease inhibitors have been designed. In the case of HIV, sipinavir is one, and lopinavir is another one, darnavir, amprenavir, and these are some of the antivirals already reported in the literature. Right, in the last 50 years, about 180 or 150 antivirals have been developed, and these are very neatly reviewed. Not only they are given as the one, the combinations of the antiviral combinations have been developed. Really very difficult task to model this. not only two drugs have been combined and three antivirals have been combined and so on. And these combinations are very, very difficult to work out. All right, people have been trying to work on this. Yeah, combinations. Right, there are a lot of combinations of the drug molecules. In the case of our work, what we did is we have taken these proteins, spike protein, the main protease, which we call it as a 3CL-Pro, PL-Pro, and RNA-dependent RNA polymerase. This we have taken. And then the list of compounds that we have taken is we have FDA approved, as well as the investigational drugs. Totally, it is about 4,000 molecules. And these molecules have been selected, and these have been interacted with all this spike protein, 3CL-Pro, as well as with the TMP RS2 and JAK kinase, right? These are TMP RS2 and JAK are the human proteins, so which are involved in the pathways, pathogenesis of this particular virus. So we have taken these, and you can see that the antiviral from this FDA phase 0 is 73, phase 1 is 10, and phase 2 63, 38 for these are the antivirals that is in various stages of trials. You can see that these are the molecules. So we took all these antiviral and interacted with all this protein, and you can understand that the value paritapravir, which shows a value of 9.3, all right, with the 3CL-Pro, and it shows PL Pro with a 7.9, RDRP 11.1, S Pro 8.6, and other non-structural proteins I am not going to discuss here. The same has interaction energy of 8.6 docking score, and with the JAK also it is 8.8. You can see that based on our analysis, the paritapravir works out to be a very good molecule, all right, for handling this disease. There are dolutegravir 9.3. This is 9.3, all right, and then nelfinavir minus 8.0, dasabuvir 7.1, raltegravir 8.1, lopinavir 8.2. Therefore, there are all these are possible. These are all the values which are greater than minus 7.5 that I have listed here. And these combinations are really good combinations. And then sofosbuvir, it's another molecule. People are talking about this molecule today for this pandemic. It is 8.1. All right, another is maricaviroic, maraviroic is 8.7. So these, if you look at this sofosbuvir, it fairly binds well with almost all that we have seen. Right, the binding energy is very high. Similarly, the maraviroic also know that binding energy is reasonably very high. So we have generated this data from the docking, right? And this database we are very clearly, very carefully we are. Galadasphere is another one. It is 6.2. People are talking about Galadasphere. It shows good activity with this, but synthetically it is very difficult to do that. So we try to get all the 100 and not three viruses and their interactions with all these proteins has been done with the 3CL-Pro, PL-Pro, RDRPS-Pro, and so on. So we have this data. We want to analyze this very critically. In this case, we have a human pathway dependent proteins, TMP, RS2, and JAK. These are also very, very important one. And then you can see that this is the active site of the protein is here, right? So these portions are really important portions wherein this ligand molecule can bind and interact, right? So this is one site and this is another site. These two sites are very, very important sites for the TMP RS2. This is serine protease domain. The another is called the SRCR domain, this domain. So we interact at the ligands in the encircled domain and we calculated the docking score and other properties. If you enlarge this, you will see that it consists of histidine and aspartic and serine. These three amino acids are there. All right, with this, we looked at the interactions. These are the details, and you can see that for RDRP, we have certain molecules, And I'm not going to the details here. We screened all this with the help of the docking. In the case of a TMPRS2, what are the molecules one can see? We have classified them into various categories. Under bronchitis category, we get certain molecules, pimectazine and indacitrol. No, they have the binding scores of 9.2, 9.28. All right, these molecules are really showing potential. You can see from the values here, right, which are really more than 6.5. Right, all these are Schrodinger based scores. And you can see that the bromoxine is minus 5.8, and you all know that it is mucolytic and it is repurposed for the COVID. Therefore, dexamethasone, it is minus 8.0, it is also repurposed and so on. This gives you some indication that certain molecule can be used for this. Under asthma category, in the Zafar Lucas is 8.4. That is another, and then Monte Lucas is 8.5 under asthma category. Therefore, you can look at the indications, therapeutic indication. Zafar Lucas is to prevent asthma, and there are indicetol is it is used to treat the breathing trouble and wheezing. All right. Therefore, there is the kind of indication that we have for the COVID and this treatment are more or less the indications that we got are more or less matching from the docking. Right. Ambrexol, it is used for the respiratory disease. Again, it shows 6.0, and bromoxine is 5.9. I have already told you. Carbocysteine is 5.2. All right, these are the less binding energy category, but there is a possibility because of the concept that I am explaining to you. So what we did is subsequently there are many molecules that we have screened. We have now library we have built for this. Let me go one step beyond this. This is the a protease catalytic triad in protease domain. You can see that this area, in the case of a TMPRS2, we have serine, aspartic, and histidine. This is the catalytic triad. In this region, we put our ligand molecule with the help of the docking. And then what the best compounds that we have selected, and subsequently what we did is we did the molecular dynamic simulations with all those drug molecules. All right, and you can see the trajectory. It is more or less, all right. Now we went up to 100 nanosecond. All right, after that we analyzed again by taking these details from the molecular dynamic simulation from the trajectory. Again, we recalculated the binding energies, right, with the approach, what is called MMGBSA method. And then again, the binding energy we have calculated and compared with the docking result. And we are trying to reframe or re-understand certain aspects of these molecules. Right. So what I do here, I stop here in the last 1 hour or so. What I have done is I have tried to give you a flavor of various techniques used in molecular modeling in understanding the drug discovery process, right? By taking certain examples. Basically, in the last part of my presentation, I took the SARS-CoV-2 and then the important protein that today people are looking at it, 3CL-Pro, PL-Pro, RDRP, and the human dependent pathway, the TMPRS2 and JAK. And then I have given you the detailed results on TMPRS2 by looking at the various FDA approved repurposed drug. And then after docking, we took all those complexes and then we did the molecular dynamics. Again, we are looking whether the what we obtained from the molecular docking is equivalent to the what value that we get from the molecular dynamics. Is there any order change? What is the level of order change? And what kind of accuracy that one can get from the molecular dynamics and so on. Therefore, we use whole host of technique that is available today to understand this, right? And I thank the organizers for this opportunity to talk to you on this particular platform. If you have any questions, I'll be happy to answer you. Thank you. And I would like to thank my postdoc, S. Kannan, Dr. Kannan, and other students for their contributions to this particular project. Thank you, one and all. Thank you for the opportunity.
Thank you, sir, for such an enlightening talk. I'm sure that all the participants would have gained uh, uh, very nice information about the molecular talking as well as QSAR. Especially your work on the COVID was very interesting. So now I will, by thanking you, I will now open the session for questions and answer. So there are a few questions already in the chat box. So I will just read out those questions. So the first question is from Geeta, uh, Dr. Geeta. So she is asking that for QSAR, is it very necessary that we select molecules from the database, which is same as the designed molecules? Or should we select molecules with certain common groups that is present in the designed molecule?
Yeah, generally, if it is a homologous set of molecules, it will be useful. The prediction will be accurate, right? But it's not necessary. But the IC50 value that you select should be from the same group or should be from the same kind of a experiment, right? Otherwise, it may not give you the values that you are looking for.
Okay. So she has written one more question connecting to that. So I have designed some molecules and did the docking of those molecules. How can I go for QSAR?
What are the information that she is having? I don't know about that. If it does not, she should have, in order to develop a QSAR, she should have some experimental information on IC50. All
All
right.
right. Yeah.
Yeah. Without that, she can't develop any QSAR.
Right. Right. So there is one more question from Ragi Sirisha. The question is, shall we rank the molecules based on their free energy of binding or Ki values during the docking?
Yeah. It's a good question, but both are related, right? Yeah. You can use free energy. Yeah. You can use free energy as well as you can use K. Both are possible. Yeah, it depends on the what kind of a model that you want to develop.
Right. Right.
They are related in a sense.
Right. So are there any questions from participants? You can unmute yourself and ask questions if there are any questions. So if there are no further questions, what we will do is we'll just quickly take one snapshot with our respected speaker. I will request all the participants to please switch on their cameras so that we can have one photograph together or the snapshot together. So can you stop sharing your screen so that we can have
Yes,
a...
yeah, I will do that.
Thank you. So it's requested that everyone please switch on your camera so that we can quickly have one snap. Thank you, everyone. Thank you, sir, for such a nice lecture. So with this, we will end this session. We are going to start with the next session immediately after this. I think Professor Ivan Scutino is already there, if I'm not wrong, so that we can start with the next session within next 2 or 3 minutes after the quick introduction. Scutino, sir,
Yes.
you are
Good
there.
day. Yes, good day to you all.
Thank you, sir. So welcome to the workshop. So I will give a brief introduction about your talk and about you, and then we can start with the next session. So let me share my screen quickly. So welcome, sir, and all the participants for the second session of this talk. I'm sorry. I'm sharing the right screen. Yeah. So our second session is on the homology modeling in drug discovery, and sir is going to explain you all about the homology modeling. Coutinho sir is an expert in computer-aided drug design, homology modeling, docking studies, pharmacomodeling, QSAR. He has done extensive work in this field. He has also expertise in synthesizing NCs, identification and dynamics of peptides, protein using multinuclear NMRs like 1H13C or 15N NMR. Sir has more than 180 international publication and one patent to his account. And he has received several awards and fellowships or memberships. He is on the board of several committees, member of research committees, RRC committees. He is also on the RRC committee of our institute, and we have been benefited from his expertise a lot. With this short introduction, I would like to welcome sir, and I would request him to start with his session. So I will now, sir, stop this sharing my screen so that you can share your screen. Thank you, sir.
Thank you. Thank you for that introduction. And thank you, Dr. Ashish, for this invitation. So I'm going to talk to you. Just a minute. Let me share. Is my screen visible? Is my screen visible?
Yes, sir, it is visible.
Yeah, okay. So I'm going to talk to you today about how protein structures are determined. I'm going to put the experimental versus the theoretical methods. Now, this is not a judgment of experiment versus theoretical. What I'm going to do is simply put forward the experimental methods and also the theoretical methods. And you can make a judgment yourself of the two techniques. Okay, before I begin about protein structure determination, I have a little saying from Greek mythology. Okay, Sisyphus is condemned to an eternity of hard labor. His labor is frustrating, fruitless, for just as he's about to achieve his goal, his work is undone, and he must start again from the beginning. So here you can see Sisyphus, actually the task that he has been given as a punishment is to roll this huge rock up a steep mountain. Actually, as he reaches the top of the mountain, it rolls down and he has to do this all over again. Now you begin to ask yourself this question, what has this to do with protein structure determination? This is the connection between Sisyphus punishment and protein structure determination. Those who work in protein structure prediction seem to share the same fate. Okay, and as we go along, you'll begin to understand the meaning of this statement. Okay, so what is protein structure determination all about? Given a 1D sequence of the protein, that is the sequence of amino acids I've written here, lysine followed by glycine followed by leucine, etc., etc. If we have this 1D sequence, how can we predict the 3D fold or the three-dimensional structure of the protein? And you know, such a protein can adopt a variety of folds, a variety of structures, but what we are seeking is the 3D structure, which has a minimum in the free energy. That is of essential importance because medicinal chemists believe that this minimum structure in the free energy surface is the one that is related to function. Okay. So here it is shown pictorially. How do I find methods that can convert this one-dimensional sequence into this 3D fold, into this 3D structure? And from this 3D structure, how do we assign a function? That is what we are seeking. from sequence to structure and to function. This is actually the holy grail of computational chemistry in terms of protein structure prediction. Okay, to convert a 1D sequence into a beautiful three-dimensional structure, as shown here, is the holy grail of structural bioinformatics. Now, what are the implications if we can do this? From a 1D sequence, if we can derive a three-dimensional structure and further to predict its function, if we are able to do this, we have a grasp over biological processes where this protein is involved. It can open up vistas into new discoveries and lead to new biological inventions. That is the advantage. If we are able to take a 1D sequence and predict its three-dimensional structure and its function, then we have a handle or a view into all of these processes. And undoubtedly, here is a gold mine waiting to be discovered. Okay, so first, let me address the experimental methods of protein structure determination. So there are both the experimental methods as well as the theoretical methods. So the experimental methods are X-ray crystallography, NMR, I should say here, ND NMR. Okay, we need 3D, 4D, 5D NMR, and also multi-nuclear NMR, proton, 13C, phosphorus, deuterium, 19F, etc. Okay, so if we are able to do NMR in multiple dimensions and for this various NMR active nuclei, then we are able to decipher protein structures in solution. Then we have FRET. And finally, mass spectrometry. I'm going to spend some time in these three methods. Then we have the theoretical methods, ab initio modeling, homology modeling, which will be the main focus of my talk, a little bit about threading. And though we have modeling on a lattice, I don't think I have time to talk about modeling on a lattice because I'll be spending an appreciable amount of time on homology modeling and a little bit on threading. And I'll put these two in context with each other. When do we use threading for determining a structure? And when do we use homology modeling for determining the structure? Okay, first about X-ray crystallography. Okay, for X-ray crystallography and for NMR, we need a physical sample. We need about between 5 to 10 milligrams of my protein. Okay, and you know, with molecular biology, we can do cloning, expression. Okay, as you can see here, cloning expression. We can isolate the protein after doing the expression, purify, then determine its structure by X-ray crystallography or by NMR. All that we require is this much amount of a pure protein. The only difference is for NMR, I need a doubly labeled protein. That is, my protein has to be labeled with 13C and 15N in order to do a 3D or a 4D NMR. How do I get a doubly labeled protein? Okay, that is while expressing the protein, okay, I need to grow it in a medium that is rich in 13C and that is rich in 13C and 15N. So my medium must be rich in this isotope of carbon and in this isotope of nitrogen. So what do? We actually feed labeled glucose. And for 15N, we will add into the medium 15N ammonium chloride. Okay, so that's the only difference while I'm doing my cloning, expression, isolation, and purification for a sample that is to be used for NMR. I must have a doubly labeled protein. 5 to 10 milligrams of the protein is sufficient. Okay, so now let's go to X-ray crystallography. For X-ray crystallography, I need to make a single crystal of my protein. And here is the biggest obstacle to studying the structure by X-ray crystallography. Now, you must have made crystals in your organic chemistry laboratory. And most of the times you have been successful in making some beautiful crystals for which you have been evaluated by your organic chemistry teacher and given marks. Unlike organic molecules, okay, proteins are notoriously difficult to crystallize. Okay, but however, if you can do this and not only make a single crystal, but also one which is stable under the conditions which X-ray crystallography is done, then you can put it in your goniometer, bombard it with X-ray, and get this diffraction pattern, this beautiful diffraction pattern out here. From this, you have to use a Fourier transform algorithm to convert the diffraction pattern into this electron density map. Okay, so a Fourier transformation of the diffraction pattern, that is the scattering, and along with the phase of each one of these waves, okay, we can convert it to the electron density map. And from the electron density map, we can actually fit a model, okay, which is nothing but the three-dimensional structure. And here is a little bit of biochemical knowledge which you need, okay, which is necessary in order to convert this electron density map into a meaningful model. Of course, after we have built a model, we always do a refinement of the model. And I'll talk about model refinement a little later on. Okay, so X-ray crystallography actually gives us a structure in this solid state. Okay, this is one thing that you must appreciate. Okay, so the structure that is determined by X-ray crystallography is impacted a lot by crystal forces. This you must keep in mind. And I'm not going to comment too much on this, except that you must remember that X-ray crystallography gives us a structure of the protein in the solid state. And as I told you, the biggest hurdle in X-ray crystallography is the obtainment of a single crystal that is stable to the conditions under which X-ray crystallography is carried out. Another big advantage of X-ray crystallography that it is not limited to the size of the protein. Proteins with very large size, for example, the capsid of viruses have also been studied by X-ray crystallography. Okay, proteins with a huge size, they can very, very easily be studied by X-ray crystallography. So that is one of the distinct advantage of X-ray crystallography. Then we come down to NMR. So in NMR, what we do is we basically take our protein, okay, and put it in an aqueous solution and then put it in an NMR spectrometer. Okay, so NMR spectroscopy gives us the structure in solution. Okay, so this is to contrast with what I said about X-ray crystallography, that we have a structure in the solid state And here we have a structure that is in solution. And very obviously, okay, many of us will then say that this is the biologically more relevant structure. All of you will agree with me that a structure in solution is biologically more relevant than a solid state structure. That is not to take away the advantages, the merit, and the value of a solid state structure. Please don't misunderstand me, okay? I'm not undermining or belittling or making little of X-ray crystallography. So to determine a structure by NMR, what are the things that we need to do? Okay, we need to record and extract the chemical shifts from the NMR spectrum, the NOEs, okay, the nuclear overhauser enhancement signals we need from the NMR spectrum to extract the dihedral angles and the dipolar couplings. So once we have this information about the chemical shift, the NOEs, the dihedral angles, and the dipolar couplings, we put this information into a molecular mechanics program to determine the solution structure. Now, there is one difference between the structure that we obtain by NMR, sorry, by X-ray and NMR. X-ray, you get a single structure. Okay, NMR gives us an ensemble of structures. An ensemble simply means a collection of structures. A collection of structures. Okay. Now, I don't have time to discuss the advantages of a single structure versus an ensemble of structure and why all this is there, but you must keep this in mind about NMR, that we get a collection of structures which are closely related to each other. This is because, okay, the chemical shifts, the NOEs, the dihedral angles, and the dipolar couplings which are extracted from the NMR spectrum are all used as restraints in order to satisfy a given structure, which means that from this data, actually we can obtain several structures which all meet this requirement. Okay, so that is why we get an ensemble of structures. Okay, so this is a second technique. Another disadvantage of NMR is that there is a size limitation in protein structure determination. It is difficult to obtain an NMR spectrum, a clean NMR spectrum of proteins which are very large in size today because of several problems. First is because NMR spectroscopy is an insensitive technique. Okay, it's an insensitive technique, and therefore we require large samples to record an NMR spectrum. And when you try to push a large amount of a protein in a very small volume, NMR spectroscopy is recorded in 0.6 ml of solution. When you try to push a large amount of a protein into such a small volume, the protein tends to aggregate and you get very broad lines. When you get very broad lines, you are not able to actually extract the chemical shifts, the dihedral angle, dipolar couplings, and the NOEs. And if you cannot get accurate chemical shifts, the dihedral angles, and dipolar couplings out here, then obviously, okay, we are not going to be able to determine a very accurate structure. Okay, so now I put into context NMR and X-ray crystallography. I've given you the advantages and the disadvantages of each other. Now let me go to the third technique, FRET, fluorescence, resonance, energy transfer. FRET is described actually as a molecular ruler. Through FRET, actually you can measure, okay, you can measure basically how different parts of the protein are in context with each other. Okay, so here is one domain of the protein, here is another domain of the protein, and FRET enables us to establish how these two domains are in relationship with each other, whether they are close to each other or whether they are far away from each other. To do this, your protein must be tagged with what are called as fluorophores. Now, these fluorophores are nothing but basically fluorescent proteins. And here are two very popular fluorescent proteins which are used to tag segments of the protein. Okay, here is a cyan fluorescent protein that is attached to my protein of interest at some point in its structure. Okay, so some part of the structure of this protein, which I call it as A, has been tagged with a cyan fluorescent protein. And another domain of my protein of interest is tagged with a yellow fluorescent protein. So here are the two fluorophores which have been put on different segments or on different sections of my protein out here. Okay, now what are the characteristics of these fluorophores? For example, the cyan fluorescent protein, okay, you can excite it at 458 nanometers, simply means that you can shine light on it at 458 nanometers, and it will emit a light at 480 to 525 nanometers. Okay, so you shine light at one wavelength and it emits light at another wavelength. The characteristics of the YFP fluorophore is that you need to shine light on it in this range and it will emit light at 525 to 575 nanometers out there. Okay, so having understood this, now let me tell you how do we measure the structure or the fold of that protein. Okay, so what must happen? Okay, the emitted light from your cyan fluorophore should actually excite the yellow fluorophore, and this happens only when the cyan fluorophore and the yellow fluorophore are within 50 angstroms. So as you can see here, I need the emitted wavelength of my CFP to overlap with my excitation wavelength of my YFP, and that will only happen, this overlap of the emitted light of my CFP will only overlap with the excitation of my YFP if these two fluorophores are within 50 angstroms from each other. Okay, if this happens, then what you'll be able to see? You'll be able to see and you'll be able to detect an emitted wavelength in this range. So all that you got to do is excite at this wavelength and detect at this wavelength. And if you can do that, you will get an idea that your protein is folded in such a way that these two parts are in close proximity to each other. Okay, so you must remember now one difference between FRET and the other techniques. The other techniques like X-ray crystallography, okay, X-ray crystallography and NMR. Both of these techniques actually give us atomic resolution, give us structures at atomic resolution. What do I mean by atomic resolution? It simply means that I can tell exactly where every atom of my protein is located in space. That is meaning of structures at atomic resolution. Every one of my atoms in my protein can be determined with very high accuracy. Where are they in space? That means where the X, Y, and Z coordinates, I can very precisely determine, okay, for each and every atom by X-ray crystallography. And to some extent, okay, to some extent, Okay, not with the same accuracy as X-ray crystallography, but with a great deal of accuracy, I can also do this by NMR spectroscopy. Recall that I said that I give you by NMR spectroscopy an ensemble of structures. So what it simply tells me that an ensemble of tells me that a particular atom 1 can be at slightly different positions in space. That is what I'm meaning by an ensemble of structure. Atom number 1 has a likelihood of being here, a likelihood of being here, and likelihood of being here. And that is what gives me an ensemble of structure. Okay, I'm fairly accurate. This distribution in their coordinates is really not very large. In contrast, as you begin to see, fret gets me basically an overall fold. You know, I can get what is called as a gauche folding of my protein. I cannot determine by fret actually the structure to atomic resolution. This is not possible. All that I can actually say is a gauche and an overall fold of the protein. Okay, so now having put forward these 3 experimental methods, I will now go on to the computational techniques. And first you will ask yourself this question, why do I need computational methods? Okay, it so happens that not all proteins can be crystallized. Okay, which means that for some proteins, I will not be able to determine their structures by X-ray crystallography. There are also some proteins which are not, whose NMR spectra are not easy to record for reasons that they aggregate in solution. And I told you, once they aggregate in solution, your NMR peaks become very broad. And from these peaks, they're not able to extract the chemical shift, the dipolar couplings, etc., which are essential. Computational methods are basically looking for the lowest free energy conformation, are looking for the structure which has the lowest delta G. Now, you all know that, okay, proteins, okay, because of their large number of degrees of freedom, can actually take on a variety of structures, myriad structures. But what we are interested is the lowest energy structure, or in other terms, which you must have also heard, is as the global minimum. Anyway, this is not easy to determine. If we cannot find the global minimum, at least we can seek for a local minimum in the structure. Okay, so currently, to do this, 2 principal methods that I use is molecular dynamics and Monte Carlo simulation. What molecular dynamics is all about, basically, okay, you use Newton's equations of motions to basically solve for how atoms move on a potential energy surface. A potential energy surface that is actually determined by an empirical force field, often called as the molecular mechanics force field. So actually, basically what we do is, for each and every atom, we compute the forces that are acting on this atom using a molecular mechanics force field, okay? And then we know that the atoms will move according to their accelerations. Remember that force is equal to mass into acceleration. So if I know the forces, I'll be able to determine the accelerations, and these accelerations will actually move the atoms to new positions in space. So I can actually solve this for each and every atom. As they move on the potential energy surface, because of the forces and the associated accelerations, the atoms move to new positions in space. And that is, they actually generate a new 3-dimensional structure. And I can keep on iteratively doing this. So once I get a new structure, after solving for the forces and the acceleration, I can repeat this. And for the new structure, again, recompute new forces, new accelerations, and keep on doing this a large number of times in order to arrive at what is called as a trajectory. So molecular dynamics actually gets me a trajectory, okay? And from this trajectory, I can use molecular mechanics minimization to actually extract local minima and the global minima. Okay, So here you see molecular dynamic simulations that has been done for 1 microsecond, a pretty time. You can see various structures that have been obtained during this molecular dynamic simulation out here. Now here is an example, okay, that was done in 1988 by the Nobel laureate, okay, sorry, by Peter Coleman, who passed away. He would have been one of the persons who would have got the Nobel Prize had he lived. Okay, you know that the Nobel Prize some years back was shared by 3 people in computational chemistry. Okay, Warshel and others. Okay, this was actually solved for a very small protein, 36. Okay, the structures were solved, but using a very long simulation time of 1 microsecond. Okay, 1 microsecond. In those days, this was the state of the art, and they achieved this with a supercomputer which had 256 CPUs. It took 4 months of simulation in order to arrive at these structures. However, today with modern technology and modern computers, we can easily do this in less than a day. Okay, with graphics, with graphics today, you know, we have accessed the power of the graphics today. Okay, and we can do this in a very short period of time. Okay, coming down to Monte Carlo. So Monte Carlo is another technique for determining protein structures. It simply starts from a random structure. Okay, you have a random structure which can be generated by a variety of techniques, and what you do to this random structure, okay, you make very arbitrary changes to any one or more atoms and generate another structure. Okay, so from here you actually now will move any one of these atoms, one or more of these atoms randomly in space, generate another structure, and then you calculate the energy of this structure. If this structure has a lower energy than this structure, you keep this and keep on doing random displacements to one or more atoms and keep on generating a lot of structures out here. And from this, basically, you can then finally do energy minimization and then gather the local minima, and more importantly, the local minima out here. Okay, compared to molecular dynamic simulations, Monte Carlo is not powerful enough to find an optimal conformation, even for simple cases, that is for very small proteins out here. Okay, so it has a problem, especially for large size proteins. You're not able to actually get the true or the accurate structure, that is a good local minima or the global minima out here. Okay, then I come down to the knowledge-based methods out here. And these knowledge-based methods use a combination of statistical theory and empirical rules in order to derive the 3-dimensional structure. The most successful of the theoretical methods is homology modeling. And I will now dwell a little bit about homology modeling. Okay, so homology modeling is based on this theory. I need one structure, at least one structure. Okay, I need one protein, let me say that, whose structure has been determined experimentally. This is the requirement to apply homology modeling to my target protein, that is to my protein whose 3-dimensional structure is of interest to me, whose 3-dimensional structure I want to determine. I need one protein belonging to the same family as my target protein whose structure has already been solved. Okay, experimentally, either by X-ray or by NMR. Okay, and homology modeling actually uses this information in order to derive and arrive at the structure of my target protein, my protein of interest. Okay, so now how accurate can we determine the structure of the target protein is squarely dependent upon this protein, which I call it as now my reference protein. If my reference protein is very closely related to my target protein and has a lot of similarity in basically the sequence, okay, if I look at the 1D sequence of my target, And if I look at the 1D sequence of my reference, if there is a great deal of similarity in my 1D sequence of my reference and my target, then the structure of the target can be determined with great accuracy. When there is a lot of deviation and a lot of difference in the 1D sequence of my reference and the target, then the accuracy in my structure determination begins to fall. And that is nicely established here in this slide. Okay, so when your reference and target proteins have similarities that range from 30 to 100%, you can get sort of, I'll put this in quotes, accurate structures. Now, what do I mean by, what is the degree of accuracy? What is the degree of accuracy? I'm going to talk about it later on. When my reference and my target protein, okay, differ in similarity from 0 to 30%, then homology modeling cannot be applied in order to arrive at the structure of my target. Then I use other techniques such as threading, also referred to as fold recognition, and ab initio methods in order to arrive at my structure, at the structure out here. Okay, so a different set of methods have to be used when my reference and my target are dissimilar to the extent of less than 30%. Okay, now let's come down to this range. Okay, as I told you that homology modeling can be applied when my reference and target have a degree of similarity that is from 30 to 100%. As you had obviously guessed, higher the degree of similarity between my reference and my target. Target is the one whose structure I want to determine. Reference is the one whose structure has already been determined by experimental methods. As these two get closer, okay, from 70% to 80% or even to 100%, then I say that the structure of the target can be determined. For the dissimilarity between my reference and my target, the accuracy in the structure starts to fall down. So as a result, okay, what can I actually do with my target protein when it is dissimilar from my reference protein has a great impact on modeling and drug design. This is what I'm going to show you in my next slide. When the sequence identity between my target and my reference, it's also called a template, is from 10 to 30%, then you cannot use this for structure-based drug design. You cannot use the structure of my target in order to do docking or structure-based drug design. No. Okay. Structure-based drug design and docking can only be done when my target and template have a sequence identity that is greater than 60%. Okay. So when the sequence identity is lower than this, from 0 to 50%, you can always determine a three-dimensional structure. You can always determine a structure. So I'm not saying that you cannot determine a structure when the sequence identity is less than 60%. What I am saying that the utility of this structure, okay, the utility, for example, in medicinal chemistry, we want to do docking after we have built a structure for my target. Okay, I want to do docking, I want to do structure-based drug design, I want to design novel drugs, novel ligands, novel entities. This is not possible if the target and the template have a sequence identity less than 50%. Okay, You get a structure, but when the sequence identity is less than 50%, what you can do is you can use such a structure for assignment of function and for site-directed mutagenesis. Okay, so I'm emphasizing this. When you have determined a structure from a template whose sequence identity is less than 50%, do not use it for docking and for structure-based drug design. All that you can use such a structure is for assignment of a protein function. And that also is useful. You'll be able to say to some degree of accuracy, how does this protein function? What its function is going to be? It's going to be like. And you can also use it for site-directed mutagenesis out there. Okay, so now you begin to understand the value of homology models. People build homology models and do not take into consideration the sequence identity between my target and the template, and very indiscriminately use it for structure-based drug design or for docking, or in order to design novel drugs and novel this. We must be careful when my identity falls below 60% out there. Okay, so if you have a fairly good homology model, then you can do a lot with it in the drug discovery process. You can use such homology models for target identification, target validation, lead identification, and for lead optimization. You can do a lot if your homology models are accurate. That is, in other words, when they have been built from templates which have a high degree of sequence identity, then you can do all of these things that is listed out here. I have no time to talk about all of this. All of this can be done if your homology model has been built from a template that has very high sequence identity. Okay, now I do not have too much of time. I would have loved to talk about each one of these steps in homology modeling, okay, and the care that we need to take in each one of these steps. Okay, there is a lot to talk about in homology modeling. I almost have 50 slides about talking about these various steps in homology modeling. However, time does not permit, but I'll talk about a little bit about each one of these steps and what care should be taken in very brief. The first thing is, when I have my target protein, the first thing is to identify to which family my target belongs to. Okay, to determine the family to which my target protein belongs. Okay, and I need at least one member in the same family whom I told you, whose structure has been determined. So the first thing is, this target, I must identify the family to which this belongs. You know, the family of proteins. At some time during your undergraduate classes and your postgraduate classes, you may have been taught in your biochemistry classes about protein families. Okay, there are protein families. Okay, so I need to identify first to find out my target protein to which family it belongs to. And once have I identified the family to which it belongs to, I need to find out one member in the family whose 3D structure has been determined. Okay, then from this member whose 3D structure has been determined, I need to identify what are the structurally conserved regions. What do I mean by structurally conserved regions? Structurally conserved regions are basically parts of my protein which have regular structures such as alpha helices and beta sheets. Okay, what is it about structurally conserved region? You know, SCRs will actually be found in all members of that family. That is what actually characterizes a family. A family of proteins are characterized by, they all have the same types of SCRs. That is, the number of alpha helices, the number of beta sheets are generally conserved within a family of proteins. Not only are the number of alpha helices and the beta sheets conserved to some degree within the family of proteins, but also the relationships between the alpha helices and the beta sheets, okay, is also preserved in this family, which means that actually the families have very similar structure. All members in the families have very similar structures because they have the same SCRs. And this is what actually puts all the members in the family together based on their topology. Okay, and this is why that I can use the structure of one member of the family in order to derive the structure of my target protein. This is the basis of identifying a family, because all members in the family to which my target protein belong will have very closely related structures, and essentially they will have the SCRs. So once I have identified the SCRs in my I can use those SCRs basically to put it the same SCRs to put it into my target to put into my target protein, to cut paste the SCRs and to structure it into my target protein. In order to do this, basically I need to align my target protein with my reference protein. With my reference protein. Okay, to do this alignment, to bring them into relationship with each other, that is to find a correspondence between an amino acid in my target protein and another amino acid in my reference protein, that is referred to as alignment. And there are several techniques that are used for alignment, and the most widely used method for alignment is the Needleman and the Wunsch algorithm that helps us actually to find the relationship between the 1D sequence of my target protein and my reference protein. So once I have done this and I know here is an SCR, that is here is an alpha helix, what I do, I cut paste this structure into my target protein. Here is my beta pleated sheet. Then I cut paste this structure into my target protein out here. So that is the value of alignment. Once I've aligned out here, then I can assign the SCRs based on this alignment out here. Okay. So now let me go back and again illustrate this, what I'm talking about. Okay. So I have now my target protein, my reference protein, and here are the SCRs. These blocks are the SCR, the alpha helices, the beta sheets out there. Okay, so what I've done, I've identified the SARs, and now basically what I've done, I've translated the coordinates for my reference protein to my target protein. I translate these coordinates and I say that in my target protein, here also is an alpha helix. Because it is present in my reference protein, there will be this part from this part also an alpha helix. If this is a beta pleated sheet, what I simply do is now I translate the coordinates of this beta pleated structure to my target protein and I say here is also a beta sheet. Likewise, I do this for all SCRs throughout the sequence of my target protein. Now, what are these parts? These are unstructured parts. I told you that SCRs are referred to as structurally conserved regions. So these parts which actually connect your SCRs are referred to as loops. Now we must determine the structures of the loop. And there are various techniques in which we can determine the structure of these small segments, okay, which connect the SCRs, which are referred to as loops. We have various techniques in order to determine that. We can actually determine the structure of the loops from related proteins. Okay, and these related proteins may not be from the same family. It doesn't matter. In order to determine the structure of the loop regions, we can take the structures of the loop regions from any protein, provided that this protein also has the same loop, okay, the same size. This sequence of amino acids here, okay, which is in my target protein, is of the same sequence in any other protein. It need not be a protein that belongs to the same family out there. Okay, so what we have done now, for my target protein, I've built the backbone structures. I've completed building the backbone structures. And now you know that basically there are side chains. Now we have to build the structures of the side chains. And we can do the side chain structures by various techniques. Essentially, what we can do is we can find in databases. Okay, for example, let's take a particular amino acid that is glutamine. Okay, and I want to determine the structure of this side chain. So one of the ways in which we can determine the conformation of the side chain is to look at proteins that are there, for example, in my PDB, and see the various conformations of glutamine that are present in structures in the PDB, and adopt one of them based on certain principles. I can adopt that structure and put it for the glutamine in my target protein. So all the side chains, basically what I can do, I can use databases in order to construct the structures of these side chains using certain principles out here. So now I've completed building the structure of my target protein. I built the backbone, I built the structures of the loop, I built the side chain conformations out here, and now I have a gross structure, a gross 3D structure of my protein out here. And from this gross structure, what I need to do, I need to do a refinement of that structure. And this refinement is normally done by any energy minimization techniques or by molecular dynamics techniques out here. So I've tried my level best to tell you the steps in homology modeling. Now, each one of these steps, there are a lot of conditions to be met, okay? A lot of precautions to be taken out here. It is not just as simple as I've just put to you in the last 5-6 minutes out here. Okay, but that is not important. Okay, at this point of time to go into so much of details. What I've tried to give you is a glimpse of how homology modeling is done. So after I have refined my structure, I actually have to do a validation for my of my model out here. Okay, so validation is a big thing today. Okay, because remember, all of these are theoretically done, and always the question is, how do I know that the structure that I have developed is not a result of some chance? Because whenever you do something theoretically, the structure that you develop can also come as a result of some. It could not be statistically sound structure. Okay, it could not be a meaningful structure. In order to show that it is sound, it is accurate, we need to validate a model. And one of the most popular tools that is used for validation of a model is what is called as 3D profiles. Okay, 3D profiles is actually used in order to validate my model out here. Okay, so now here are all of these steps, and how all of those 7-8 steps, what are the intricacies of these steps? I do not have time. Okay, organizers, can you just tell me how much time I have left? Hello, Dr. Ashish? Hello? Yeah, how much time do I left? Yes? How much time is left? I can't hear you. It's echoing.
Professor, could you have another 15 minutes?
15 minutes?
Yeah.
Oh, so I rushed through my... Okay.
Yeah, can take another 10 minutes. It's okay.
Okay. Okay. Okay, so actually there is a lot of theory to cover, on homology modeling. I think I will skip this, and I will talk a little bit about how do we determine structure when the sequence identity falls below. Okay, this part, when the sequence identity falls below 30%. Okay, when the sequence identity between my target protein and my reference protein is from 20 to 30%, then the method that is used is not homology modeling. Homology modeling cannot be used in order to determine the structure of my target protein. I use a method which is called as threading or profiles. Okay, now I'll spend a little time about what is threading or profiles. Okay. Okay, so here is about threading or profiles. Okay, the first thing is like homology modeling. I need to do an alignment of my target protein with my reference protein out here. Okay, so that needs to be done out here. Okay, so here is my target sequence. Okay, and what I'm evaluating is basically the environment in which my sequence is found. Okay, I'm going to determine. So I've already established a three-dimensional structure of my target protein for my reference protein out here. And this is the three-dimensional structure. So I now look at the environment of lysine. Okay, the structure has been determined. So lysine has a particular three-dimensional structure. And I look at its environment. Now, what do I mean by its environment out here? So I look at how much of lysine is exposed. How much of the lysine is buried? Okay, what is the local structure of lysine? Whether lysine has an alpha helical structure or beta helical structure out here. And based on this, okay, various environments have been classified. Okay, based on the degree of how the amino acid is exposed, how much of the amino acid is buried, what is its secondary structure. We have various environments out here. Okay, so now I actually look at the compatibility of lysine with its environment. Okay, compatibility of lysine with the environment. Now, what has been done? A lot of study has been done, and it has been known in a variety of protein structures that lysine likes to have a particular environment. Okay, so let me go back and say a little bit thing. Okay, based on this, how much has been exposed, how much is buried, and what is the secondary structure? We have 6 types of environments: P1, P2, right up till E. 6 types of environments have been located. Okay, what is the difference between the 6 types of a P1 environment has a certain percentage of exposed, a certain percentage of buried, and a certain type of a secondary structure. A P2 environment has a different percentage of exposed, has a different percentage of buried, and a different secondary structure out. So we have these 6 different environment types. Okay, now what is done is people have looked at databases and have seen that lysine prefers one of these environment types. Let's say lysine, in most of the times, okay, when we look at protein structures, we often see lysine likes to adopt a P2 environment. Okay, now for my target protein, I have built a structure, and now I have found that lysine in my structure has a certain type of environment. And let's say now in my structure, lysine has been put inside an environment P1. Actually, lysine likes a P2 environment, but my structure prediction method has put lysine in a P1 environment out here. So you see that there is now a little incompatibility. Now you can say that your lysine structure has not been accurately determined. So there is a method of actually doing this compatibility between what lysine likes and what lysine has been found in that particular in its particular environment out here. So I have a compatibility score. So in my 3-dimensional structure, it seems that a compatibility score of my this is leucine, sorry, not lysine. Sorry, this is leucine. Lysine is this. Okay, this is leucine. I'm very sorry. L is leucine out here. Okay, leucine. Okay, has a compatibility score of minus 0.7. Not a very good compatibility score. On the other hand, proline. Okay, proline has been put into an environment which is its natural environment, and the compatibility score here is 0.5. Okay, so you see that the structure that has been determined for my target protein, when I do this compatibility scores with its environment, I get these various scoring numbers. And then I add all these scoring numbers and I get a total score of minus 0.3 out here. Okay, that this number now, which is the score of each one of these amino acids in their environments, these scores give me an inkling or a feeling of how good my protein structure has been determined. A negative score tells me that actually my protein structure has not been determined very accurately. There are a lot of amino acids which are not in their native environments. Okay, the structure that has been determined has put a lot of amino acids in not native environments, not in the best environments that they would actually like to be in. Okay, so if I get a score that is in the positive range and a greater the value, that tells me that basically, okay, the target sequence, okay, the structure has been determined with a fairly good accuracy, that every amino acid has been located, has been positioned in one of its best native environments out here. Okay, so this is what threading is all about. Actually, after we build a structure, we begin to assess whether every amino acid in that structure actually adopts a compatible environment, an environment in which it is comfortable in actually. And the score actually tells us about how each amino acid is in its environment, the compatibility of each amino acid in its environment. A positive score and a greater the number, okay, this is 0.3, greater the number, okay, tells you that the structure has been very satisfactorily determined out here. So this is about threading, which is often done for, okay, sequences where the target and the reference have a sequence identity that is less than 30%. Okay, below 10%, we have to use ab initio methods, that is physics principles, in order to determine the structure out here. Okay, so I think, okay, so let me give you one example of homology modeling. Okay, here is an example of homology modeling. So remember, I told you the use of homology modeling, whether you can use in structure-based drug design. Hello, there is some disturbance. I'm going to conclude in another few minutes. I'm going to conclude. Here is an example, okay, of an inhibitor which has been designed based on a homology model of human rhinovirus type 2. This is an important target for the SARS-CoV-2 disease. Okay, so HRV2C is an important target in SARS-CoV. The structure of this target was determined by homology modeling, and from the homology model, this inhibitor was actually designed, okay, and it has come into clinical use. Okay, so we can use homology models in order to determine novel leads and novel compounds, provided that the structure has been determined from a template which the sequence identity is high, as I told you, from 60 to 100% out here. Okay, so I've talked to you a little bit about homology model, though I've not spoken in great details about each one of these steps. I just try to get you a feel. Okay, I just will end with, one of the structures that we have built using homology model, and its structure is here. This is the structure of an protein for tuberculosis, that is a methionine transferase protein. It's an important target in tuberculosis. Okay, we have determined the structure using homology model. Okay, we also developed some inhibitors against this, theoretical inhibitors. We did not go on to actually synthesize some inhibitors against this MTB-MAT protein. Okay, so I'll conclude with my talk out here, and thank you all for your patient listening. I know I couldn't do full justice to homology modeling, but I hope I've given you a brief and the value of homology models and when it can be used and what care you need to take when you're developing homology models. So thank you for your attention.
Thank you very much, sir, for such a wonderful presentation and explaining us about the homology modeling and what are the advantages of homology modeling over the X-ray crystallography and NMR technique. So this session is open for question and discussion. So if anybody is having question, then either can unmute and ask the question or can post in the chat box. So meanwhile, I have one question.
Yes.
If we carry out the threading, if we prepare a model using threading methodology, and if I follow a paper which has reported the threading methodology, protein structure, so can I use the active site which is predicted by that report, or should I work on it on myself?
As I told you, threading can get you basically a grosh fold. Okay, so you have to be careful if you are trying to infer the active site. You may be able to do this. Okay, you may try to infer the active site, because remember, by threading, you do not get a very accurate structure. Okay, it's not a very accurate structure. So be careful if you are trying to infer too much about the active site. You can infer about the function. Okay, but going down into details about trying to infer what is the active site and trying to dock compounds into the active site, it is a no. Do not use such structures for docking of a structure-based drug design. You will get very wrong answers. All that which you can use safely is to determine that this protein has this sort of a function. This is how it will work. There you'll be very safe. Okay, but using that for medicinal chemistry purposes, like for docking of a structure-based drug design, or trying to understand the structure of the active site and seeing what are the amino acids that are there inside the active site, etc., you must be very careful. Okay, you could be right, but I say that there are greater chances of making errors in trying to extract too much information about the active site, the structure of the active site, and the details about the active site. Be very careful. Safely, you can predict its function.
Okay. Right. So it means if we don't have proper structure, so we should not enter in the structure-based drug design. Instead of that, we can prefer ligand-based drug design if the ligand structures are available.
True. Very true. Very true. The sad thing is that I see a lot of people who have built homology models with very poor sequence identities with their templates, with their reference proteins, and gone ahead and tried to do docking and structure-based drug design out here. That is a big mistake. Use docking and structure-based drug design only when your sequence identity is very high. To be on the safe side, I would say between 70 to 80%, then you are in a safe zone.
Okay. Okay. Thank you very much, sir. Any other question from participants? Okay, so we'll conclude this session here. Thank you, Kutino sir. Once again, thank you very much for sharing your valuable knowledge with all of us. Thank you everyone. So we'll start our next session at 2:30. So I request everyone to join back at least 5 or 10 minutes before the next before the time for the next session. Thank you everyone.
Good afternoon, everyone. We will start this session in next 2 to 3 minutes. So good afternoon, everyone, and welcome to the last session of today's workshop. This session will be conducted by Dr. Ashish Kanet on the demonstration of Autodoc software for molecular docking. So before he starts with the demonstration, just let me introduce Dr. Ashish. Dr. Ashish is working as assistant professor at SVKM's NMIMS SPPSPTM since last around 2 years. He obtained his PhD from University of Baroda, and after that he worked as a research associate and senior research associate at Naveen Saxena Research and Technology Private Limited. He had also worked as a postdoctoral fellow at University of KwaZulu-Natal in South Africa, and after that he, I think again worked he gained more experience at the industry before joining at SPPSPTM. His research expertise include development of design and development of new chemical entities for different diseases, including Alzheimer's disease, tuberculosis, hypertension, etc. He is also interested in the reaction setup using continuous flow reactor, but his main expertise is in the computer-aided drug design work, which he is going to explain us today with the help of molecular docking using Autodoc software. So with this short introduction, I would like to request Dr. Ashish to start the session. So I'm going to stop sharing the screen, Dr. Ashish, so that you can start with your session. Thank you.
Okay, thank you, Dr. Kapil. So we'll start with some computational techniques where I will explain how to use the Autodoc software. My screen is visible to everyone, right?
Yes, it is visible.
Okay, thank you. Okay, so I'm not going in much detail about the introduction part because in the last 2 days we already have discussed a lot of theoretical concepts. Okay, so I'll directly jump to molecular docking. Okay, so what is molecular docking? When we allow two molecules to interact with each other, and when they find a particular orientation, or what we can say, when they find comfortable orientation with each other, that we can say as dock structure or molecular docking. Okay, when these are the chemical structures, then we call it as molecular docking. And generally, traditionally, what we can say, we consider non-covalent interaction as molecular docking. But nowadays we are also studying the covalent interactions or covalent bonding analysis in case of drug receptor, or we can say ligand receptor interaction. So here the knowledge of the preferred orientation, that means something is getting oriented in some other molecule or with other molecule. That means we can say the ligand is small molecule and the receptor is a bigger molecule. So the ligand is interacting with the receptor structure, and when ligand is interacting with the receptor structure, both are changing their orientation, or we can say the conformations. Okay, so what we can say here is the orientation, particular orientation of that molecule depends on their preference, or we can say that compatibility with each other. Okay, and the fall in energy or the drop in energy when two molecules come close to each other is considered as docking energy. Okay, when there is high energy, the molecules won't come together or won't show any type of interaction. When there is drop in energy, then only we can say that molecules are interacting with each other. Okay, so the success of docking depends on two properties. Number one is search algorithm and another is scoring function. Okay, so there are different types of search algorithms, which include systematic algorithm, molecular dynamics algorithm, genetic algorithm, Monte Carlo. Yes, Monte Carlo algorithm, which Putino sir and Subramaniam sir also have explained about this. But we'll see, we'll briefly discuss this part as well. So what is search algorithm? When particular molecule exists freely in space, then that molecule has freedom to orient in any possible way, I'm using word possible, any possible way. But when it is interacting with certain other molecule, then what happens? It tries to get specific conformation, which is in that conformation that molecule is comfortable, okay, with itself and as well as with the receptor structure or the other molecule. Okay, so identifying that particular conformation or identifying particular orientation of the molecule, it comes under the search algorithm technique. Okay, so the next is scoring function, that is nothing but force field. Now what is force field? Force field includes or consider different atoms and their bonding with each other in the molecule. Okay, each atom, because of its electronic nature, shows different properties, and their bonding with other atoms makes those atoms to behave differently. For example, if any electronegative atom is connected with neutral atom, then there will be electron transfer. That electronegative atom will be pulling certain electrons from the neutral or some electron, I can say electropositive atom. So such type of properties are getting changed. Okay, so what force field does, it is mathematical calculation which determines which type of atoms are there and which types of bonds are there in the structure, and calculates the energy of the structure, total energy of the structure. Okay, and when we carry out or when we perform molecular docking study, the receptor and ligand, generally we talk the macromolecule and micromolecule. We can say macromolecule, here we can say the ligand molecule or small drug-like molecule, and the macromolecule we can say the protein or genetic material. Okay, so that comes under the scoring function, right? So when interaction happens and the energy obtained is in the negative term or the lower value is obtained, then that is considered as better interaction. That means there is fall in energy. Okay, and that interaction energy is considered as docking score, and we generally measure it in terms of kilocalorie per mole. Generally unit of the measurement is kilocalorie per mole. Okay, so these are the sampling algorithms, different algorithms followed by which are being used by different softwares. So matching algorithm is there, which based on the three-dimensional orientation of the molecule, or we can say the geometric based uh, algorithm. It considers the orientation of the particular molecule, and from that molecule it tries to generate different conformation. Okay, and this type of algorithm is preferred by these all softwares. Then incremental construction is there, where fragment based uh, what we can say fragment based interaction is performed or carried out, and different types of conformations are generated. Means one ligand molecule is there, different fragments are made internally, and those fragments are allowed to interact in different pockets of the active site. Okay, and depending on the scoring of those fragments, the entire molecule is again considered, and that specific conformation is used. Okay, similarly, MCSS is also works on the same principle. Then LUDI is there from the BioVia, which also works on the same principle. Then Monte Carlo is there, which is stochastic approach, which generates random conformations. Means if you have specific conformation at X moment of the time in the space, then it randomly converts that conformation into another conformation. There is no logic in the changing or converting one conformation into the another conformation. Okay, and then comes genetic algorithm. This Monte Carlo was used in the Autodoc versions of the Autodoc, that is Autodoc 3 or before any version before the fourth version. A genetic algorithm is there, which is based on the Darwin's theory of evolution, where similarities and differences between two conformations are identified, and those similarities and differences are used to generate the new conformation. Okay, so this is basic principle of the genetic algorithm, which is being used in the different softwares. So here, okay, so molecular dynamics is there. Molecular dynamics is a little bit time consuming, which requires high performing computers. So generally these type of uh, conformation generations are not preferred at initial level, but if you want to analyze the structures in flexible form. For example, in general, what we do in the docking, we consider receptor as a rigid molecule and we consider ligand as a flexible molecule. But in case of molecular dynamics, we take receptor as well as ligand, both as flexible. And because of the flexibility in both the structures, interacting structure, it consumes a lot of time to carry out or to perform or to identify the different conformations with respect to time. Okay. So, Autodoc, if we talk about the Autodoc generally nowadays Autodoc Vina is in more use. And there is Autodoc tool, Autodoc 4 and Autodoc Vina. Two different tools are there developed by same group. Okay, so what is major or basic difference between these two tools? The major difference is the scoring function. In Autodoc, the scoring function is based on the Umber force field. Okay, and in Autodoc Vina, it is hybrid scoring function, that is empirical plus knowledge based scoring function. We can say that MMFF, that is Merck molecular force field, is there, which is being used in the Autodoc Vina, or we can say that generalized force field GFF is there, which is being used in the Autodoc. Okay, so and one more difference is that Autodoc 4 is more manual approach, and Autodoc Vina is little bit what we can say is command based, or we can say that if we give one or two commands, it carries 4-5 steps easily. So Autodoc Vina is much faster, and it depends on the structure. It is more accurate. What type of structure we are providing, the accuracy of Autodoc Vina is, we can say that it is more accurate as compared to normal Autodoc or Autodoc 4. But obviously at the end, it depends on the structure, which type of structure which we are studying. Okay, so in Autodoc, there are different types of files are involved. or we can say in any type of docking, few files are required. The PDB, that is protein data bank file, is required, which we know, or majority of us know, that this type of file we can obtain from RCSB, that is protein data bank, which is freely available on the Google, which we will visit in few moments. Then for Autodoc, there is PDB QT file is there. Protein data bank, this PDB stands for protein data bank file. This Q stands for partially charged file, and this P stands for specific atom type. There are specific atom types which are involved in Autodoc. On the categories of those atoms, the charges are given to the structure, and that's why it is called as PDB QT file. Now there is grid parameter file is there, and grid log file is there. Grid parameter file is something is which we manually generate, where all the information of grid is saved. Okay, and the grid log file is there, where whatever experiment we are performing on the grid parameter file are getting saved, or uh, getting recorded in the grid log file. Okay, this is generated when we perform certain experiment on the grid parameter file. Similarly, docking parameter file is there when we carry out the docking. Then certain we carry out certain mathematical expression on the docking parameter file. Then we get that information or the result in the docking log file. So these types of files are involved in the, what we can say, in this type of software. Okay, so now how to download, from where to download this Autodoc and how to install it. So I'll briefly inform you all about that. So just a minute. Okay. Okay, so for installation of Autodoc, you need two files. Okay, one is MGL tools. You need to download and install MGL tools, which you can download from the MGL tools, which is Scripps Institute, which is from the Scripps Institute. You can download it from here. Okay, depending on your system, you can download. Unfortunately, it is not available for the Mac, or we can say Apple products, because that Apple product is using the Catalina OS, that is newer update, and for which the software is not yet developed. Okay, so if you are using Mac product, then you may not use this software directly. You can create virtual box and then you can install it. That is different thing. Okay, so you can download it from here, and then the next is Autodoc tools. So you can visit this Autodoc tools website. स्क्रीन सबको दिख रहा है ना? Okay, so from this Autodoc tools site, you can go to this download section, and you can download the Autodoc 4.2. You need not to perform this step, registration step. You can directly proceed to the download section. Okay, so once you scroll down, you'll find product specific link. It's the rather than saying product specific, you can say that operating system specific link. So depending on your operating system, you can download this file and you can install. Once you install the MGL tool and Autodoc tools, you can visit your C drive. In C drive, you can find program, go to program files, right? In the program files, if you scroll down in alphabet T, you will find one folder, the Scripps Research Institute. The Scripps Research Institute. Okay, so when you double click on this, you will find, if you have installed Autodoc, you will find this Autodoc folder. If you have installed Vina, you will find this Vina folder. So we will talk about Autodoc right now. So double click on Autodoc. Again, there will be one folder with the version. So click on it, and in that you will find these two application files, Autodoc 4 and AutoGrid 4. Okay, so today what I'm going to do is, I'm not going to take a receptor and ligand and show you how they are interacting in Autodoc. I will tell you what are the problems one face during performing these molecular docking studies in Autodoc. Okay, and we'll discuss or we'll try to troubleshoot those problems, because many times what happens, many people or many researchers, many students try to get Autodoc from the website and they try to run it. They know the steps, but they fail some at some or the other point because of some errors. Okay, okay. So in today's session, we are going to deal with that part only. Okay, and this Autodoc 4 and Autodoc Vina, we are not, we cannot cover these both these softwares in this given time slot. So we'll focus on the troubleshooting part more than discussing how which steps should be followed after what step and all the about that software. Okay, so in case of this program files Autodoc, there are two application files. Okay, these are main, or we can say that these are important files to perform Autodoc. So what you have to do is just select those files and copy those files. Do not make any changes in these files. You can, if you try to open these files, you will find different lines in those files. Okay, these are certain mathematical, or we can say that programming is there on which programming the software runs. Okay, so do not try to edit those files, otherwise the programming will fail, or you can say the process will fail. So just copy these two files, and you can paste it somewhere else in your working directory. Okay, so let me show you. So I have created a directory on the desktop. Instead of using word directory, I have created one folder on the desktop. Okay, and I have pasted these two files here. Now whatever uh, programming or whatever interactions I am trying to do with the receptor and ligand, I will do in this folder, and I will use these two files. I will not disturb those two main files. Okay, so this is one thing. Now next, let's visit to RCSB side. Okay, that is protein data bank side. So I hope that downloading and installation is clear to everyone. So this is protein data bank site website, where the protein structures which are generated experimentally and which are identified by either X-ray crystallography or NMR technique. Okay. Today in morning session, the Coutinho sir discussed this part, how to generate protein X-ray protein structure, three-dimensional protein structure by using X-ray technology, and how to generate a three-dimensional protein structure using NMR technique. Okay, so all such developed three-dimensional structures are deposited here. The coordinates of such structures are deposited here. So how you can search a protein, particular protein here is, you can just type your target name here, or if you know the protein code, then you can type it here, but let's start with the target name. Okay, so let's look for the acetylcholinesterase. Okay, if you visit the Wikipedia page, okay, you'll get lot of information about this acetylcholinesterase. And if you look on at this right hand side section, you'll see here the three-dimensional structure of this protein, right? So if you further scroll it down, you will find uniprot. Okay, uniprot for human, uniprot for mouse. Okay, so this is protein university. You can say that uniprot is protein university of proteins. There you will find all types of proteins, at least sequence of all type of protein in this data bank. Okay, if you scroll up again, you will find the PDB, and you can get two links, PDBE and one is RCSB. So you can access the three-dimensional structure from RCSB, or you can go to the uniprot site. Okay, I have selected the human acetylcholinesterase. Okay, the role of acetylcholinesterase is hydrolysis of acetylcholine, which is involved in various neurodegenerative diseases. So we'll not go in that part. So once we are at the uniprot site, what we can do? Click on this blast. Blast is nothing but similarity identification tool. Okay. So we have selected blast. We will find here the amino acid sequence. 1 letter amino acid sequence of your entire protein is displayed here, right? So below that box, you'll find target database. Which target database you want to analyze or you want to evaluate. So I want to evaluate human database. Okay. So I'll click on the, or we can, let's take the three-dimensional PDB database, not select specific species. Let's select the three-dimensional PDB, that is protein data bank structure, and just click on the run blast. Okay, it will take few seconds to find out the similar structures or the similar sequence from the protein data bank. If you don't know the code, then we can use this technique. Okay, so you can see here alignment, different alignments are mentioned over here, and the entry query is mentioned over here. Okay, so on this basis, let me click on the first alignment. Okay, you can see here 100% matching is there. Okay, that means there are several three-dimensional structures are available. So let's go back to the site, just copy this name and paste it in the PDB structure and click search. Okay, so you will find here there are 833 similar type of sequences having three-dimensional structure, which are from the Homo sapien, that is human species. Okay, there are different species which are mentioned over here, and the numbers are given in front of that. This much protein structures are deposited in the protein data bank. So now you'll find it very confusing which structure to select, right? There are so many protein structures given in the protein data bank. Now, which structure should I select as a starting point for the computational study or for the docking study? Okay, so in that few points you should look first. One is the most important method point is method of generation of that structure. You can see here X-ray diffraction is there. You may find somewhere NMR technique. Okay, and nowadays cryo technique is also there. X-ray, and we can prefer cryo also, but NMR technique one should not prefer if X-ray and cryo structure is available. Okay. Now next is organism. Okay. You can see here Tetronatsi californica. T. californica organism is mentioned here, which is not obviously, which is not Homo sapien, which is not human. Okay. So you can scroll down and try to find out the Homo sapien, or you can just click here and filter. You can select the filter from this. You can click on the Homo sapien, so you will get all the Homo sapien structures here. But at same time, you can consider the protein structure from this organism if they are showing maximum similarity. Okay, similarity in terms of amino acid sequence, type of amino acid. These are 2 points where one should look at the similarity. The third point where one should look at the similarity is the properties of amino acid. For example, 2 amino acids are not matching. For example, the one particular amino acid from this organism and particular amino acid from the human, which are appearing in the same position in the sequence. By type they are not matching, but if they are matching by property, then third type of similarity is by property matching. If they are matching by their properties, if acidic in nature, if aromatic in nature, or if non-polar in nature, if basic in nature, these type of properties, amino acid properties are there. You can find this classification on Wikipedia page of amino acid. So if they are matching in that way also, then that is also fine. So this T. californica and human sequence for acetylcholinesterase is almost same. Okay, so once you are clear with that, the next point one should look is the resolution. First is method, second is species, then third again come back to this method, the resolution. Okay, with what resolution the receptor structure is resolved. That means lower the value is considered as higher resolution. If you find here 2.5 and in somewhere if you find 3, that means 2.5 is much better than 3, because lower the value means higher the resolution. Okay, so this resolution one get from when we actually try to get the three-dimensional structure of the protein. When we keep our protein sample or protein crystal under X-ray crystallographic technique, the X-rays we allow to bombard the X-rays to interact with the protein structure, and depending on the electron density present in each atom present in the amino acid of that protein, the X-ray get diffracted from the structure. Okay, and depending on the angle of the diffraction, psi and phi angle of the diffraction, the electron density present in that particular amino acid or particular atom, rather than saying amino acid, particular atom, the electron density is identified, and on the basis of electron density, the atom is assigned. For example, if 14 is there, then we can say that nitrogen is there. If 16 is there, the electron density value comes to 16, then we can say that oxygen is there. In the same way, we can generally we go further, and in identifying that, this resolution plays very important role. Okay, nitrogen and oxygen. Okay, if you look at the atomic number, they have 7 and 8 atomic number, there is only 1 unit difference. If we don't have proper resolution in our technique, then the density for nitrogen may we may interpret the density of nitrogen as density of oxygen, or we may misinterpret the density of oxygen as density of nitrogen, and we may assign the wrong atom. Okay, so for that purpose, the resolution is very important, and selecting the proper PDB structure from this protein data bank, one should also look at this resolution value. Okay, so on these 3 points, one should look at this. Select the structure. So I know the specific code, so I'll directly go to that specific code. Okay, this 4-digit specific code is assigned to each protein data bank structure or PDB structure. So if you know the, or in majority of the publication, you will find PDB code, okay, which is nothing but this 4 alphanumeric or 4-letter code. Okay. So this is one ACJ is also acetylcholinesterase. This 2CKM is also acetylcholinesterase. So once you enter in any specific code, so you'll find, if when you scroll down, what you will get, the chains present in that structure. If you find chain A, that means only one chain is there. That means this is monomer. Okay, acetylcholinesterase enzyme, monomer form. If you scroll down further, you will find different types of heteroatoms present in the structure and different types of ligands, or we can say drug molecules present in the structure. Okay, so here you can see this AA7. This is bistacrine. Okay, let's explore this structure. Yes, you can click on the ID. So you can see this is one tacrine molecule. This is another tacrine molecule. These 2 tacrine molecules are connected by a linker or spacer. Okay, so this molecule is present in the active site of this receptor, right? So okay, this much of information is there. Now how to download this protein structure in 3-dimensional form? So once you are inside this specific code, there are 2 options: display files and download files. So go to download files and click on PDB format. Okay, so you'll get that PDB format downloaded here. Okay, now again, what we can do is scroll down and look at the ligand. Click on the ligand structure or ligand code. Okay, and you can download that ligand from here in SDF format, structure data file SDF. So it will appear in certain values. Okay, so you can copy this, you can open textpad and just paste it over here. Okay, then file, save as. Okay, yes, I'm giving any random name, save. Okay, this is one way. The another way is just copy all this information, okay, and use the software which we have discussed yesterday, that is OpenBabel. Okay, then change this input, or rather than interfering that input below. Okay, input this ignore this information and use the following information. You can choose here SBF. Okay. And you can convert that into any sort of information. Just a moment. I got a call. Okay, so this SDF we can convert it into any sort of format, either MOL2 or PDBQT or PDB. Okay, just select any sort of format and just click on the convert button. You will find here the molecule is converted. Okay, so what you can do here is just copy this information. Paste it over here in the text pad, and I'm just clicking on the save button. Just let me close it. Okay, I have saved it on the desktop. Okay, so now let's go to properties. Okay, will not go in that part right now. Okay, so this is one other way where from which we can convert the molecule into PDB-QT. Now we can save that file at particular positions place. Let's say a.pdbqt. Okay, and just click on save and click again on the convert. Now you will, if you look at that file folder or the location, you will get a.pdbqt. That means the file from the specific position which we had downloaded in the SDF format, we have successfully converted by using OpenBabel into the Autodoc required format, that is a.pdbqt format. Okay, again you can cross check it. You can open it with textpad and you can see here different atom types are mentioned over here. Okay, charges are mentioned over here. Okay, and the XYZ coordinates are mentioned over here. Okay, so the initially the charges are 0 because there were no charges from the original structure, but any time of point we can add the charges. Right, so this is second way by which we can convert the structure into the required format. Now let me discuss one more method of converting the structure into the required format. Okay, so if we don't have specific file, what we can do is just open any drawing software. Marvin Sketch. Let's check with the Marvin Sketch. I think it is open here. Sorry. Yeah. Okay, so you can draw any structure, which we already have discussed in yesterday's session, how to draw the structure. So I will not discuss that again. So go to File, Save As. Okay, and you can save in any format. Okay, so I prefer Trapos CB or Trapos Mol2 format, and I'll click save as. So you can again go to your folder and check it whether that structure is saved or not. You can see here mol2 file is properly saved. Okay, so there is one more software. Just a minute, please. Sorry for disturbance. Yes, so by these many ways we can convert the structure into the required format. Now there is one more tool which is very interesting and which is very important is Avogadro. Avogadro is freely available. You can download it from Google. Okay, so you can draw structure here and you can import structure here as well. Any way is possible. Just go to file, you can open and select, you will get structure in the working space. Okay, what is important here is you can add hydrogens if missing over there. You can remove hydrogens if you don't want that original hydrogens, you can remove and you can build it again. Add hydrogens. Okay, then okay, there are different force fields are there. You can minimize the structure. Okay, and there are, this is crystallographic part, will not go there. So different force field are there. These are empirical force field, MOPAC, Gaussian, James. Okay, these are empirical force fields which you can use to minimize the structure or to optimize the energy of the structure. And then again you can go to file, save as, and you can convert extension into any format. Okay, so let's jump to the Autodoc tools. So once you install the Autodoc and what we can say MGL tool, you'll find such icons on your uh, installation location or the desktop. So you can open Autodoc tools. Okay, so this is what Autodoc tool, and you can do majority of the experiments here itself without using any command prompt. So I'm not going to run entire job here because that is not possible to perform in the this time slot. So we'll discuss certain important points over here. So once you are completely installed, properly installed Autodoc, you can go to File. The most important thing is you need to change the path or you need to set the path. Okay, so go to Preferences and Set. So this is startup directory. So at the first moment, this will be the your installed location or installation folder. So you can change that directory to any other directory. So this is my desktop folder. So what I will do here, I'll just click on this path location. I'll single click here, so it will show me the entire path. So just copy it from here and come back to your software. Remove this original path, just paste the newly copied path and click on set and make default. If you click on make default, for your next job also, this folder will be taken into consideration. So if you want to perform new experiment or new jobs in different location, then again you have to change this location. So this is very important. Otherwise, randomly your files will get stored at random places, and at next step or at in the docking step or in the grid generation step, you will find difficulties. So just click on set, make default, and click on dismiss. Okay. So now I want to import a protein structure here. So go to file, read molecule. So it will direct me to the my folder only. It will not take me to the installation software installation folder. Okay. So if I don't find PDB structure here, I can look at this extension if it is correct or not. I'll change the extension. Okay, there is no stored PDB, so I'll download it from internet source. Okay, I've downloaded it from internet source, that I will copy it to my folder. Okay, so go back to the software. Now you can see here in your working directory, there is one PDB structure available. So click on it, click on open. Okay, so you'll find this protein structure. Now, there's so many water molecules are there which we do not require. Why these water molecules are here? These water molecules are got trapped while developing crystal structure of protein. When we express protein in particular organism or particular cell and we try to crystallize it out, water molecules and certain salts or heteroatoms get trapped in the protein structure, which are actually not required for our study. Okay, so we can easily delete out these water molecules. So what we need to do that, there are different ways. I'll tell you few ways. So if you click here on this arrow, this will expand the code and you'll find there is only one chain. So again, if you expand this chain, you'll find different amino acids, right? So keep on scrolling. Okay, so now here you can see these are the water molecules. So one way is I can select one water molecule at a time and I can delete, that is time consuming. The other way is directly I can delete all the water molecules. So instead of looking at this left part of left panel of this software, I can go to edit, delete water. So just you can look at the screen. Once I click this delete water, all these dots, red color dots will disappear. Okay, these are the oxygens, hydrogen suppressed. This is hydrogen suppressed graph of the structure. Okay, hydrogen suppressed means hydrogens are not visible. Okay, so these red dots are oxygen, where hydrogens are there but not visible. So just click on the delete water and you can see all those dots are disappeared completely from here. So now when you expand this tree from this left panel and you scroll it down completely, you'll find amino acids and few heteroatoms. Now what are these heteroatoms? Just click on this square and it will get highlighted in the window. Okay, so you can check it here. So there are two NAG molecules. If you take cursor over here, you will get complete name of that. There are two NAG molecules and there is one AA7 molecule. Now, what are these molecules? If you don't know, go back to the RCSP site and look at the structure again. So AA7 is the ligand, that is also non-protein atom. NAG is also non-protein atom which is present over there. Okay, so I want to delete this NAG, so I'll select it. I'll select another NAG. So I selected two NAG. Okay, so selecting this square is enough. That means these are selected, and you can see here in the working area the two parts are highlighted with yellow dots. So go to edit, delete, delete selected atom. Edit, delete selected atoms. It will ask you whether you really want to delete. Then if you are sure, then delete it. If you have selected wrong entry, you still have one chance to go back and correct it. So I'll click on continue. Now you can see these two parts are disappeared from the working space. Now I have ligand over here. I have selected that, and you can see here it is in this region, and this is the active site of the receptor. Okay, where the ligand is present. Okay, I don't want ligand either. Okay, so I'll go to edit, because I don't want ligand in this form. I want my own ligand to be docked at this position. Okay, so how to identify the grid box and how to prepare that grid box, that few techniques I'll explain, and from where you can easily get the idea about how to generate a grid box. Okay, so select this part, go to edit, and again go to delete selected atoms. Continue. So this ligand is disappeared from this structure. So what we are doing, we are now preparing our protein for molecular docking. So what we did till now is we deleted water molecules, we deleted any hetero molecules which are not required, and we, along with that, we also deleted the ligand. We don't want ligand right now with us here. Now next step is edit atoms, sorry, edit hydrogens and add. Okay, try adding hydrogens anyway found non-bonded atoms in 2CKM. Okay, so it is saying that non-bonded atom is there, so don't worry about that. We are going to fix that part also. And many times, many students or many researchers face one problem over there. The Python script shows you an error. So how to deal with that, that also we'll discuss. So just accept it. Okay. So right now I have added all the hydrogens. Okay, so next step is check for the correctness of this protein molecule. I hope you are understanding. So next step is check for the correctness of this structure. So how to do that? Go to edit, miscellaneous, miss is there. Check for missing atoms. Edit, miscellaneous, check for missing atoms. So click on that. You'll find that certain part of these amino acids or entirely these amino acids are missing from the structure. Okay, so select all residues. You will find yellow dots at certain positions in the structure. That means those amino acid positions are selected. If amino acids are not there, positions are selected. If amino acids are there, these amino acids are selected. So dismiss it. Again go to edit, miscellaneous, repair missing atom. Now we want to repair those missing atoms. So click on it. Depending on the number of residues, it will take some time to repair. If you are lucky enough, you won't get this error, but majority of the time people will get this error. Okay. So in that case, what to do? We cannot proceed as it is. So there is alternative way. Okay. So just close this script. Now go to Google. So this is our protein that we know. So look at the FASTA sequence. Okay, go to display file, click on the FASTA sequence. What is FASTA sequence? This is amino acid sequence, nothing but amino acid sequence. Now we have this amino acid sequence open with us. Now open new tab. Go to Swiss model website. It is swissmodel.xpc.org. Okay, start modeling. Click on this start modeling. Okay, copy this entire amino acid sequence. Okay, I'm not telling you the regular procedure how to perform molecular docking. I am telling you how to troubleshoot the errors which we are getting in the molecular docking with the Autodock software. So copy this sequence here and paste it over here. You can copy just this amino acid sequence, or you can copy with the indicator, that means this code as well. Okay, so immediately it will turn into some different color code. Okay, so click on the right. So click on the build model. It will take some time depending on the templates. You can see here, depending on the number of similar type of structures present in the protein data bank, it will take some time. Okay, while it is computing this part, we'll discuss which type of charges one should select. Okay, and how to identify the location. Okay, let's consider this is our correct protein. Okay, let's consider this is our correct protein. So go to, now I'll discuss how to adjust the grid box at particular site. Go to grid box. You'll see grid extremely sorry. You'll see the grid box appears at certain random position. Okay. Majority of time it happens, your structure is appearing at North Pole and you will find grid box at the South Pole. So how to get back it to the position? There are different ways. You can scroll these wheels and change the position or move this grid box. Okay, this majority of you might be knowing this option. So there is one more option, deja vu. Okay, so deja vu is there. So it will open one window where from you can check with the root and we can transform. Now you have you have opened this window and you are performing certain operations on the screen. So it is that operations are being performed with the whatever systems are present in the screen, that those operations will take place with all those systems. So now what you can do is deselect this root and just click on the miscellaneous. Okay, so now you can see here whatever operation I am performing, it is with this grid box. So I can move it. Okay. Again, go back to root, arrange the orientation of the structure. Okay. So instead of moving these wheels for longer period of time, you can use this shorter way, that is deja vu, and you can adjust ligands, proteins, anything by using this tool. Just you need to deselect this, mouse transformation, apply root object only. So you can deselect this, and after deselecting this, you can select whatever you want to move in the workspace. Okay, so with this you can bring the box at particular position. This is one second way. First way was by rotating these wheels. Okay, now if you know the active site, you can bring that box at particular active site position. Okay, and how to get those coordinates, that I will discuss. There are two different ways. Those two ways we'll discuss in detail. But before that, I want to discuss with you, if no reported active site, or if we don't know where is the active site present on the entire protein structure, then what we can do, we should or we can go for blind docking. What is blind docking? Considering entire protein as active site, performing molecular docking study is known as blind docking. Okay, so you can increase the size of this box by rotating these wheels, or what you can do is more easier way, just left click on this wheel, you will find one window will pop up and you can change value over here. Okay, so this is I changed with that. Here again I will change the value, apply. Okay, okay, so like this I can increase the size of the box. Again I can use the deja vu to reposition the box on the structure. Okay, so role of this deja vu is enough for me right now. So I'll use small refinement I can perform by using these wheels. So I can cover this box. Okay, so you can see here maximum value is 126 and it is covering almost majority of the protein structure. Sorry. Now, when you prepare this box over the receptor, this box says that whatever ligands you are including or you are allowing to interact with the receptor, those ligands will interact with the amino acids present in this box. Okay, so entire protein now is acting as our active site, and we can carry out the molecular docking with the entire protein. Now we can allow our ligand to interact with each and every possible site present in the receptor, and on the basis of their interaction scores, they will get ranked, they will get arranged in particular sequence, and we can identify at which region the interaction is more, at which region is interaction is less, and we can decide which is the particular possible active site for present in the structure. This is called as blind docking. Okay, so the next point here is spacing, angstrom. The spacing is mentioned in the angstrom. Okay, 0.375. Now what is that 0.375 spacing? Okay, so this 0.375 spacing suggests us this box contains n number of points. This box contains n number of points, or we can say that n number of probes are there. Okay, and those points or probes are spaced at this particular distance from each other. Okay, if anyone from you have performed the 4-dimensional QSAR study, then you might have come across different types of probe atoms. Nitrogen is one of the probe atom. Hydrogen, we can use carbon as one of the probe atom. We can use calcium, oxygen, sulfur, any type of atom we can use probe atom. Generally what we use in triphosphate or in Schrodinger or in Autodog also, we prefer carbon as probe atom. Why? Because majority of the atoms present in the protein are carbon atoms. Okay, so we can take as a carbon atom, but in this software we don't have any choice to change that probe atom. But if you try to perform manually the 4-dimensional QSAR, you will get n number of or multiple options where you can select different types of probe. Generally we prefer carbon or we prefer nitrogen, because there are many nitrogens present in the protein structure, because proteins are amino acids and amino means nitrogen is there. So we can select that nitrogen as a probe atom as well, which gives us more refined interaction results. Okay, so we can increase or decrease this. Depending on that, the box size will change, because the space between two probe atoms is changing. If we increase the space between two probe atoms, then box size will increase because the number of probe atoms will remain same, but we are increasing the inter probe atom distance. That's why you may find here the change in the box size, right? So if you increase the inter probe atom spacing, then accuracy will decrease. If you decrease it, then accuracy will increase, but computation time will be much higher. Okay, so you can keep it 0.37 or 0.38, which was the standard. Okay, so this was the second point in this one should take care of. Once you are done, you can go to file and close saving current. That means this information with XYZ coordinate will be saved now, but we discussed till now for the blind docking. Now we'll discuss if we know the active site present in the structure. Okay, you remember that I'm just closing this part so this box will disappear. You remember that we have deleted ligand molecule. So that ligand was present in this protein structure, and that ligand was having certain XYZ coordinates. Okay, that means three-dimensional orientation in the space. Okay, so how to identify that space? Okay, you can look at the amino acids which are present around the ligand, which is very hectic job, though that is possible, and that can be considered as last option for identifying the active site, but that is possible option. So that is one way to identify the active site. The another way is there is one free tool, Discovery Studio Visualizer. Okay, this is from BioVia software, so it is freely available. You just need to provide your email ID and you can download it. Sorry, so you can, okay, there is registration form. It will ask you professional email ID, but you can put Gmail, Yahoo, or any of your personal email ID, and that also works. So you can download it. Just when you put it here, your email ID, and just click on next. It will go to the download section. And once you install that, you can see here the Discovery Studio visualizer is there. So now we'll get the XYZ coordinate, the second way which we are discussing, how to find the XYZ coordinates. So this was the first time installed on this system, so it is asking for some optimization or yes or no. So just click on OK. So when you first time install the software, it will try to optimize the software performance on the basis of the system which you have. It will take hardly some time. So now your software is ready to work or use. So what we can do here is just go to file, open, that 2CKM, and you will see here the protein structure is in the workspace. Now you can see here this highlighted part is your bistachin or the ligand. Now we want to know the position of this ligand over here. Just a moment. Okay, so you have selected the receptor structure, the ligand structure from the protein, and in define and edit binding site, when you go to this define and edit binding site, there are three options. From receptor cavity, if don't have receptor ligand structure here, it will try to identify the cavities present in the structure, and it will provide us the sphere. Okay, from the PDB site record, the PDB file, in the PDB file, the coordinates are mentioned. From there also one can identify, and from the current selection, because we have selected, we know the ligand is, our ligand is this one, or this is the active site, so we have selected the ligand and we have used this option. So now there is one sphere, so just select that sphere, right click on that and check for the attribute. Okay, so now here you can see XYZ or XYZ coordinates are there. You can copy it, or you need not to copy, but you can just keep this window on, go back to your software, go back to grid box. Now look at this position. The grid box is at particular position. Now I am going to change this XYZ coordinates. Okay, points. Okay, let me copy it one by one so I can paste it directly. Now you can see the box is moved little bit in particular direction. I'll select the second option, the second value, that is y-axis value. And go back to the software, just paste it. Again, you'll see the box has moved to certain position. Need not to worry, because we have not entered yet the Z coordinates or the Z coordinates. So again, copy it, go back to the software and paste it. Okay, just a minute. Let me read the molecule again. Okay. Select the ligand from current selection, right click, attributes. Yeah. Okay. Let me tell you why this is happening. So select the ligand structure from this left panel. Edit, delete water. So such type of errors happens in the Autodoc, that is major problem. So at that time, just a moment please. Okay, so here this job is done, right? So we can scroll it down. You can see here 100% sequence similarity. Okay, so the template is different, but the 100% sequence similarity. So it has generated same model. So go click on this model, download PDB format. Okay, so we'll open that PDB in the software now. File, read molecule. Okay, so now you can see here the receptor structure, two units are there, chain A and chain B. Okay, these are homodimer. What we have supplied to this software, Swiss XPASI, we supplied the amino acid sequence. Majority of the acetylcholinesterase appear in the dimer, homodimer. That means two parts are same. Okay, so we can delete out one part from it. Okay, so just select it, edit, delete selected atoms, continue. Now we are left with only one, and these are the heteroatoms that also we don't want. So go to edit, delete, continue. So heteroatoms are also deleted. Okay, so this is grid. This is our receptor. Then go to edit. Now here, once we obtained our receptor molecule from the Swiss model, we need not to correct the atoms, which we were trying to correct it from the repair missing atoms. So that error will not face here. So now directly what we can do is we can add hydrogens, where we can select polar hydrogens only and click on OK. Then add charges. Now there are Coleman charges are there and Gastinger charges are there. When to select this type of charge? Coleman charge is for amino acids and Gastinger charges are for the non-amino acids as well as for amino acids. So whenever we are dealing with the ligand or any heteroatom or any metal, or we can say any non-amino acid part, we can go for computing Geiger charges, or we can, for protein, we should select for, or we should go for Coleman charges. So you will see here total Coleman charges added 4. Okay, that is fine. What is next? Go again to charges and check total on residues, whether the charge is properly distributed or not. Majority of the time what happens, many groups, what they do, they select the Coleman charges and they do not check whether the charges are properly distributed or not. So everyone should start practicing this, check total charges. So no residues with non-integral charges formed. This is ideal message. If this message appears, then we can say that charges are distributed over all the amino acids properly. Why this happens? Because we have generated this model from the Swiss model. If you would have taken it from the RCSB, charge distribution may get varied, and then in that case what we need to do is, if there is no proper charge distribution, there will be one window which will suggest us, or which will inform us which amino acids are not having charge distribution. It will list out 3, 4, 5 whatever amino acids are there which are not having any proper charge, and below that list there will be an option distribute charge properly or something like that option is there. So you just have to click over there and the charges will get distributed properly. So this is how one can add the Coleman charges in the structure. Okay, so now next step is grid. So select the macromolecule. Macromolecule is open in the structure, so select the macromolecule. Again, there is some error, sorry for that. Okay, so you won't face such type of error when you are doing it properly. So let me tell you few more troubleshootings. So here, yeah. Just a minute. Okay. So this is one way where from you can identify the XYZ coordinate. The third way is, let's discuss the third way. Go to your folder, open your protein structure in any editor. You will see different amino acids, this number of amino acid, name of the amino acid, and the XYZ coordinate of those amino acids. For example, let's consider in my active site, I have amino acid 17. I'm just taking as an example just to let you know how to calculate that. Amino acid 17, amino acid 20, and amino acid 21 is there in the receptor active site. So the third way is just look at the 17th number amino acid. So this 17th number amino acid is glycine. It is having 3 atoms. Okay, nitrogen, carbon, carbon. Okay, hydrogen suppressed class. Okay, so select the XYZ coordinate from the last atom of that amino acid. You can open Excel sheet over here. Okay, just copy it one by one. Copy it one by one in sequence, that is XYZ sequence. Okay, so this is for 17th number amino acid. Yes, right. Let's consider another amino acid. Let's consider 19th. So look at the last atom of this arginine and select the XYZ coordinate. Why we are selecting last atom only? Because these are the terminal atoms. So let's mention it with its code, and let's take one more to understand it clearly. Let's take 21. So look at this. This is proline. Let's look at the last atom over here. Sorry. And paste it over here. So let's consider these three are the active site amino acids. So now what we need to do is we can go either way. We can select some of these three. Okay. And then what we can do is we can divide this value with number of amino acids which we have selected. Okay, so you can find here, this is the average or the center where you can put the grid box. Okay, the other way is, you know that to take an average, we'll get the same value anyway. By using any option, you can identify the coordinates of the grid. Okay, so these grid coordinates one can use while generating grids. In the grid box, we can paste it over here to get the grid box in the system. Now I can't see where is my grid box, but let's select those values and put it here. Because that grid box is somewhere in the space. Okay, so when you save this file properly, your grid box will appear at that particular position. We have not saved this file in the PDBQT format. That's why the grid file is not appearing here, but I'm sure that once you save it properly in the PDBQT format, the grid box will appear over here. Okay, so then you can go to go here to the file and close saving current. So the grid box will be saved properly. Okay, so this is what about Autodoc and troubleshooting in the Autodoc. Then we can generate the by using this information we can save the grid file, and similarly we can save the docking parameter file, and then we can run auto grid and Autodoc. Okay, so this is what about something some important information about Autodoc. Let's discuss about Autodoc Vina. Okay, when you try to carry out virtual screening, Autodoc fails there, at least on the Windows system. If you have Linux system, then also there are certain tricky operations you need to perform to get the virtual screening results by using Autodoc tools. Okay, so if we, if you want to carry out the virtual screening, let's say 50 molecules, 100 molecules which you have designed, so what you can do is you can run it by using Autodoc Vina. That is easiest way. So let's discuss about the Autodoc Vina. If you have Windows 10 operating system, what you can do is just go to store, that is Windows Store, Microsoft Store. Okay, in search you can type Ubuntu app. Okay, so this is virtual Linux. You can easily install in few minutes or in a half an hour on your system. You need not to carry out any major operations or major changes in your current operating system. Okay, so for Windows 10, it is freely available in the store. You can use it here. I already have installed, so I will not again download it. Or if you are not downloaded or installed it, one option will appear here, install or download. So from there you can install it correctly. So let me start that, and let me tell you about how to use that software. So this is Linux operating system on my Windows system. Okay. There's something going on. Yeah, I'll take these questions once I explain this part. Okay, so one green line or blue line, whichever color line will appear here, that is your login. So just what you need to do is sudo apt get update. Okay, just one when you are when you are accessing this Linux after some time, so it is always better to update it. It will take hardly some time, and it will ask your login password of your system. Just type that password, and depending on your internet speed, it will update your environment, working environment. So for me, it won't take much time because before starting the session, yesterday only I updated the system, so it won't take much time. Okay. So next command is sudo apt-get upgrade. So any upgradation of your supporting softwares or whatever softwares are associated with this Linux will get upgraded. Will get done in short period of time. We take at least 1 week to teach these softwares to our undergraduate students. So I know that this is not possible to explain each and every point in 2 hours of time, but I'm discussing major points or the troubleshooting points. And whenever you face this type of errors, you can select one or other method to overcome that problem. Okay. So if you have Windows 10 operating system, you should go for this Ubuntu Linux system on your Windows 10. So you can see here my Windows 10 is here. I can access both the system together. I can perform any Linux job, whichever I want to perform on the Linux, and simultaneously without switching operating system, I can perform whatever jobs I want to perform on the Windows system. Okay, so it has done it. So next step is I want to unlock Vina here. So for that, I will install OpenBabel in the Linux environment. I have OpenBabel on my Windows environment, but I need that software on the Linux environment. So we'll do sudo apt install OpenBabel. So already have installed it. So it is saying that the software is already there. The newest version of the OpenBabel is already installed over here. If it is not installed, it will run some commands. Automatically, it will run some command. You need not to do anything. It will ask you once permission, yes or no. So at that time, you have to click Y on your keyboard and press enter, hit enter. So entire job will run for you. Okay. So I have done this installation. So let me check where I have correctly installed OpenBabel on my Linux environment or not. So clear it. So to check whether the OpenBabel is correctly installed on my Linux environment or not, I'll just type one command o babel space -h. Okay, this is information of the OpenBabel which is present on my Linux environment. So you can scroll up and you can see here the software, the OpenBabel is correctly installed on my system. OpenBabel converts chemical structure from one file format to another. Okay, this is its introduction of that software. Okay, let me clear it again. So I'll get back to the original position. Now I want to install Autodoc Vina. So for that, type command sudo apt install Autodoc-Vina and hit enter button. It is saying that I already have installed. That's why it is saying that already the newest version is present in your Linux environment. I want to check. So just type Vina and you can see here the receptor. I have not mentioned the command for Autodoc Vina start with this Vina, and whatever you want to import from your folder, you need to type it there. That is, for example, xyz.pdb or xyz.sdf. Okay, so that you have to type in front of that. So that's why it is saying missing receptor. That is nothing wrong in that, because I just have typed here Vina, and you can see here all this introductory information of this software appears here. That means your installation is successful. Now you can use here Autodoc Vina as well. Okay, there are so many tutorials available on the Autodoc's website where commands are mentioned, which type of command to follow. But there are very less, or I think there are very less information is available how to install and how to use the Linux system in the Windows system. So I thought to share this information with you all. Okay, with this, I think we should stop here, and if we have any query, we'll take that. So any questions are welcome to discuss. I can see few questions here. How to know the heteroatoms that present in protein are drug or coexistence ligand? Okay, so what you can do here is go to the RCSB site. Okay, when you open specific protein, you can scroll it down. The chain information is there that we have discussed. Now ligands are mentioned over there. The molecule which is drug molecule or the active ligand, or just the unwanted ligand, that you can identify with this external ligand annotation box. The small below this ligand box, there is external ligand annotation box is there. So here the reported biological activities of those molecules are mentioned. If you find the information over here, that is AA7 is having IC50 of 1.5 nanomolar, then you can say that the AA7 is your active ligand or the drug-like molecule or the drug molecule. And other than that, all are the non-drug-like molecules which are present in the structure, which you can easily delete out, identify and delete it out. I hope I have answered properly to this.
Sir, one question.
Yes, please.
I had tried to use this Autodoc, but on my personal computer. Is it necessary that it has to be an institutional computer?
No. You can use it on any system. You can use it on your laptop, desktop. It is not required that it should be connected with the institute system.
Sir, please send the recordings so that we can practice it.
Yes. We'll share the YouTube link
Yeah.
after this session. how to validate molecular docking results.
Yes, yes, that is what I want to do.
Yeah. Before that, I will take the questions in sequence. So I answered the first question. Let me take the second question. Okay, that is only the second question. How to validate molecular docking results? Yeah, this is very important question where majority of the uh, students make mistake, because at the initial step we might not be knowing all these points. So please listen carefully how to validate molecular docking results. There are mainly 2 to 3 important points which one should consider. So first thing is grid validation. To validate molecular docking result, you need to validate your molecular grid which you have generated. Now, how to validate that grid? Once you have generated a grid box with your XYZ coordinate at particular position in the receptor, what you need to do is you need to identify the co-crystallized ligand. Let me share my screen again. Just a moment. Yeah. Okay. So this is specific protein has drug molecule in it. Now, how exactly this drug molecule is present in the crystallographic structure? What happens when we try to express the protein in the cells or in the microorganism? What we do, we add small concentration, or we can say that specific concentration of drug, either agonist or antagonist, that depends on your target. So we add specific amount of drug. So for acetylcholinesterase, tacrine is there, donepezil is there, memantine is there, galantamine is there. So these 4-5 molecules are available in the market for acetylcholinesterase as inhibitors. Okay, so we add particular drug molecule in the biological system, or that assay process. Okay, so this drug molecule will go to specific site of the receptor and it will interact it over there. Okay, once it interact, where it will interact biologically, at the most active site of the receptor, or at the specific binding activity site of the receptor. Okay, and when we crystallize out that protein from the our culture, the drug molecule will remain at that specific position. Okay, and when that drug molecule remain at that specific position, the conformation of the receptor or the protein, the amino acids which are present around that drug, slight change in the conformation will appear over there. Okay, please listen carefully. So slight change in the conformation around that ligand or that drug molecule will appear over there. Okay, and that decides the cavity, the dimensions of the receptor cavity. Okay, that decides the dimensions of the receptor cavity. Now, when we take out one crystal from our expressions, biological assays, and we study it under X-ray crystallographic study, it also suggests us that ligand molecule. Okay, you can, we already have seen that ligand molecule or the drug molecule is appearing in the structure. In this way, that drug comes in the actual structure. Okay, so the position and the orientation of the drug, which is biologically inhibiting that receptor, is get trapped in the X-ray crystallographic study, and the receptor cavity is oriented accordingly. Okay, so such type of ligand molecule or drug molecule which are obtained in the three-dimensional crystallographic studies are called as co-crystallized ligands. Okay, so what we need to do is, once we generate grid, take out these co-crystallized ligands from the structure, separate it out, and once we generate the grid, redock those co-crystallized ligands. Okay, if our grid is proper, that means it is correctly generated, the co-crystallized ligand will orient or get bound in the active site in same fashion as that of the original three-dimensional crystallographic structure. The ligand will appear in same orientation or same conformation, and that you can cross check by overlapping the original co-crystallized structure and redock co-crystallized structure, and you can find out the RMSD value. The root mean square deviation or the difference between the two orientations will suggest you that how exactly your redocked molecule is fit in the receptor active cavity. Okay, so this is one validation where the RMSD value should be minimum. Okay, if you are using same ligand, then the RMSD value, if it is less than 0.5, then you can say that, or you can consider that the receptor grid is correctly generated. Okay, second thing, the second important thing, which is most of the people don't know, is which type of ligands you are trying to dock. Okay, for example, I have designed few tricyclic molecules against the Alzheimer disease, which acts and inhibit the acetylcholinesterase enzyme or protein. So my design molecules are tricyclic molecule. Okay, so look at the PDB structures. This ligand is also contains tricyclic system. So the cavity, the orientation of cavity is convenient or suitable for the tricyclic molecules. But for example, let's take donepezil. The donepezil molecule is somewhat linear structure which does not contain such tricyclic molecules. So it is linear structure. So there are certain PDB structures available for acetylcholinesterase in the protein data bank, which also contains donepezil molecules. So it depends on which type of structures you have. The general topology or general appearance of the structure should be similar to the co-crystallized structure, or the co-crystallized structure with whatever our PDB we are selecting. If it is not always possible, but if it is having certain appearance similarity with our designed molecule, then we should go for that. Okay, so in that case, we'll get accurate grid generation, and our the results will be most accurate in that case. Okay, so this is another way. The third most important way of validating our grid, receptor grid, is once we carry out the molecular docking study, look at the score. Okay, score you may find -12, -14, and you may find it by number. It is very nice score, no doubt. But along with that score, look at the interactions. Your receptor is active site is there, which contains certain amino acids, and your ligands are there. Ligand is there, which is fit in the receptor active site. Now, look at the orientation of that ligand. Look at that orientation of that ligand and check that whether the ligand is properly oriented with the proper site. For example, aromatic ring is there, it should appear in the hydrophobic region. Okay, if polar group is present in the ligand, it should appear along the side of the polar amino acid. If it is not there, then there is something wrong in your grid. So by these three ways, you can validate the grid. So I hope I have explained all the questions. I have answered all the questions. So if any other question you can ask, or Dr. Kapil, are you there?
Yes, I'm there.
Yeah. So if there is no question, we can conclude here.
Yeah. So I think thank you, Dr. Ashish, for a nice demonstration on the molecular docking using AutoDock. With this last session, we would like to conclude the workshop, two-day workshop on the drug discovery. I hope all the participants have gained some knowledge related to drug discovery and using different tools. We had a hands-on demonstration yesterday on some of the chemistry softwares, and today we had hands-on demonstration on AutoTalk. So I hope that all the participants have learned something new which they can apply in their own research. And with this, before we end, I would like to thank all the participants for attending all the session and being engaged by asking questions to the speakers. So on behalf of the school, I would like to thank everyone. Dr. Mayur, would you like to say something before we end the session?
No, I just wanted to say that I hope there were a lot of students, and I hope they have benefited from this. And if anyone wants to get in touch with us, you are most welcome. We are always available to help out. So the mission was basically to train the youngsters, I mean, like, and give a good exposure. That was our intention. I hope people can take advantage of it.
Yeah, thank you, sir. So before we end the session, I'm putting here the feedback link for the day 2. It is requested that all the participants who have attended all the sessions today, please submit the feedback. The feedback form will be open for at least 15 to 20 minutes, as well as from the request from all the participants. I'm also providing links, YouTube links for both day 1 and day 2 sessions, so that if anyone wants to go back and have a look at all the sessions, they can do that. So here I'm pasting the links, YouTube links for day 1 and day 2 sessions, so that whenever you want, you can visit and have a look at those sessions. So thank you everyone for attending patiently all the sessions. And
can we request everyone to turn on cameras?
Yeah, we can take one more photograph finally. So a snapshot. So it is requested, those who are still there, request you to turn on your camera so that we can take one final snapshot with all the participants. All right, thank you everyone for being there. Wish you a nice evening, and hopefully see you again when we organize some more sessions like this. Have a great day ahead.
Thank you. Thank you, everyone.
Thank you very much. So please do submit the feedback. It will help us to improve further. And with this, I will close the session, and I'm going to end the meeting. So please copy down those links for the YouTube if you want to have a look at those sessions again.